this webinar. I would like to extend my greetings to the head of Pharmacy Department, Dr. Rerum Naturalium Pharmacy Sabtono Hadi, and our distinguished speakers, Professor Alfi Khatib, and Assistant Professor Dr. Awi Sukarni bin Muhammad Sabre from Kuliah of Pharmacy, International Islamic University, Malaysia, or IIUM. Today's webinar is the second part of the PKKM webinar series, and we will have two exciting topics. First is the metabolomics in herbal science, the study case of Orthosiphon stamineus, Momodica caratea, and Psychotria malayana, presented by Professor Alfi Khatib. The talk is scheduled at 10 to 10 a.m. And then the second topic is 3D printing, an emerging technology in pharmaceutics. Our Assistant Professor Dr. Awi Sukarni bin Muhammad Sabre, scheduled at 10 to 12 p.m. As you may notice, each topic is going to be presented for about 100 minutes, and I will remind the participants to turn off your microphone during the talk. To make sure the talk runs smoothly, in the case of accidental voice of your microphone, then the host will turn your microphone off. And we thank you for your cooperation for this thing. And if you have any questions at any time during the talk, please type them into the chat box, and I will ensure they will be brought up during the panel discussion at the end. Otherwise, please ask directly to the speaker after the talk concludes. Another important point is that please ensure you fill your attendance form that will be shared in the chat box. Now that we are ready, we will enter the first lecture from Professor Alfi Khatib in the metabolomics in herbal science, the study case of Orthosiphon stamineus, Momodica caratea, and Psychotria malayana. Professor Alfi Khatib obtained his Bachelor of Science degree from Bogor Agricultural University. In Indonesia, he was then awarded the TALIA scholarship from the Dutch government for his master and PhD in pharmacognosy in Leiden University. He also won the Dutch New Venture competition round 2 based on his PhD work on iso-alpha acid purifications in 2006. He continued to postdoctoral position before moving to University Putra Malaysia in 2008 as a senior lecturer. Afterwards, he joined the Department of Pharmaceutical Chemistry, Kuliah of Pharmacy at IIUM. In March 2013, IIUM then awarded him the Excellent Research Achievement Awards for several consecutive years from 2017 to 2022. His research on the standardization of natural products using metabolomic approach and purification of bioactive compounds, especially from Malaysian plants, has led him to securing several research grants from the Malaysian government. Until now, he has published more than 130 articles in peer-reviewed international journal that has manifested in H-index of more than 30 international patents. It is an honor for us to have you here, Prof. Alfi Khatib. Now the floor is yours.
Okay. I think I have some problem with my audio system. Sorry for that. Okay, this is my time.
No worries.
Yeah. Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. I would like to thank to the organizer of this international webinar series. Yeah, this is my second talk. Yeah, on this series. So I'd like to thank to Dr. Erna as the moderator and also the organizer of this event. Also Dr. Sabtono Hadi. I also would like to thank for the invitation, and also would like to thank to all the participants who would like to hear my second lecture. Okay, let me share my screen. Can you see my slide?
Yes, all clear.
Okay. Okay, this is my second lecture regarding the metabolomics in herbal science, So I'm sorry to the Dr. Herna, because I think I need to remove one of the plants, which is the Otosipon. The thing is, because if I include that, the time is not enough. Yeah. So I just focus on these two. Yeah, two plants, which is Momordica carantia and the Satria malayana. Okay. All right. For the introduction, yeah, why I go to this research, yeah. So my work is mostly on the standardization of the Malaysian plant, yeah. And then the approach that I use is metabolomics, yeah. So actually, I did my master and part of my PhD in the isolation, yeah, of the bioactive compounds. And then later on, I found that this approach is quite interesting, yeah, and very potential for me for the natural product chemistry, yeah, in order to identify the bioactive compounds and also to analyze, yeah, the bioactivity of the extract and also the pure compounds. Okay, the background here I show you regarding the nutraceuticals, because this herbal science, this herbal extract is really related to the nutraceuticals. Yeah, so nutraceuticals actually is the hybrid of the nutrition and pharmaceuticals, right? And then also the idea on this is by the Hippocrates, yeah, philosophy. Let your food be your medicine and your medicine be your food. I think this one is really relevant to our life now. Yeah, so that's why I think the herbal extract in this case is really in line with the philosophy of this nutraceuticals. Yeah, and then also, yeah, this product now is become emerging market. Because why? Because people awareness to use of these nutraceuticals more for the prevention than cure. Yeah, now, yeah, but now they are towards to the cure as well. Although nowadays is more to prevention, but there is the trends, yeah, to use this nutraceutical not only for prevention but also for curing. Okay. And then the awareness for the natural substances is also increase nowadays, yeah. And then they want to shift, yeah, from the purely synthetic substances, especially for drugs. Yeah. Now they want to use only the natural substances, and then not only that, they don't want any individual pure compounds anymore. They want to be more to the complex system. Yeah. So that's the things is why nutraceuticals become the preference, yeah, for people worldwide. Yeah. And then you see here the increasing of the market. Yeah. Now the OMSET value per year is more than 150 billion US dollars. So very high. So I think this will be our trends in the future. Yeah, not only for prevention, but later on after we standardize, we have succeeded to standardize the herbal, and also to find out the scientific proof, then it can become the for the curing, yeah, for the treatment of the disease. Okay, so this is actually the history, yeah? The galenicals, yeah, the Galen's formulation. The meaning is that the standard preparation containing one or several organic ingredients, which is actually is the containing of not only one plant extract, but can be several plant extract. And they are at the time really apply the holistic approach. Yeah. So they are not using the to the pure chemical substances, which is the isolated approach. Yeah, because at the time there is no technology as well to get the pure compounds. But anyway, at the time they use the holistic approach. Okay, and then after that, then they change to use the purified compounds. So most of pharmaceutical company now use only a single compounds. Yeah. So now there is another trend again. They want to come back to the previous trend, using the holistic approach, which is the thing is nowadays when we use the single compounds, problem is they cannot treat the disease, but they only can reduce the symptom. So the idea of this is we call as the holistic approach. Yeah. So this is what happened now. Yeah. From grocery stores to the drug stores. Yeah. So now you find that the nutraceuticals are available in the grocery store, not the drug store. Yeah. But this is not part of the integral part of the therapy. Yeah. So why? Because there are kind of the many adverse reports against the nutraceutical. What is this happen? Because of the lack of standardization, lack of toxicology study, lack of the pharmacokinetic and pharmacodynamic data, and lack of dose response and interaction studies. So that's why now we want to going toward this approach. Yeah. There is difficulties on this, because why? In the herbal extract, there are so many compounds, and very difficult for us to analyze one by one. Very difficult and also very expensive. So that's also a reason, why we are having difficulties, for the control the quality of the product. Yeah. So how can we solve the problem? Yeah. So how to solve the problem is by metabolomics. Yeah. By metabolomics. This is one of the way how to solve this problem. All right. Okay. Isolated versus the holistic approach. Okay. Yeah. So actually in plants there are many metabolites, or compounds, and they work synergies or antagonists among them. Yeah. So you can find that maybe when you isolate the compounds, that compounds when they are alone, the activity will be different. The activity will be different compared to the one which is in the plant extract or plant matrix. They will be different. So I have noticed for several times in my experiment, I know, and I think this is very, very obvious. Yeah. And also, sometimes if you isolate, you find the compounds become not active. But when they are in extract, can be active. Now I will show you the data on this one, right? And also antagonism effect. What we want is that toxicity. Yeah. So if you mix the compounds, the compounds before it can be toxic, but when you mix with other, it can be non-toxic, or even the compounds become helpful, become functional for our give the positive effect to our body before it's toxic. So very complicated, right? So that's why, in order for us to explain, scientifically the traditional use of medicine, we cannot solely using the isolated compounds. We need to think about when they are in the plant matrix, when they are in the extract. Yeah, but this is very difficult job. How to do that? Yeah, so that's why, and again, the metabolomics is one of the way how to solve this problem. All right. And then reductionist can be called as the isolated approach, holistic, reductionist holistic. So if we see, if you see the whole plant, this is like whole plant, then holistic. But if you see part of this only, then your analysis result will be biased, may be different from what is happened, from what is the original data, So the activity can be different when you are already isolate the compounds. Not only that, also when you analyze the compounds through the in vivo, you collect the blood, for example, and then you only analyze only one compound, for example, glucose level. Then your analysis will be different compared to the one that you analyze whole metabolite. Then it will be different. Of course, because you see from different angle. If you see from only one part, then your analysis can be biased, right? But the things, the actual one is not only one part. The actual one is whole body, whole metabolite. Okay. So again, that's a reason why metabolomics is important in this case. So then, what is metabolomics? Actually, metabolomics is the comprehensive analysis of the whole metabolite, in the sample. So if your sample is herbs, then whole metabolite in the plant extract. If your sample is the biofluids, for example, from blood or from the urine, then we are talking about the metabolite, in the whole, in the blood or also urine. Okay. So actually, this metabolomics is they are like a young technology, following the previous technology, which is proteomics and genomics. So we are not independent from the system biology. From the system biology, we have genomics, we have proteomics, and lastly, we use the metabolomics. Okay. So this is what happened long time ago. I did my master and part of my PhD, tried to isolate and find out the pre-compounds. I also get some fractions. When in the fractionation, then you lose the bioactivity. The crude extract, for example, is very, very active. But once you did the fractionation, you lose all the activity. Why? Because there is synergism effect. At the stage, you may change your title, right? Change your research topics, or change your plan because you lose the activity. This is the plant extract activity or research. Then will never be proceed anymore. Is always a secret forever, right? So how we solve this problem? We need to know the compounds which is active in the plant extract, not after the isolation. Okay. Okay, I think I already explained. You are a complexity not only in the herbs, but also complexity in the human body. The mechanism is also complex. So now we have two sides now. Your sample herbs is complex with a lot of metabolites, and they can work synergism or antagonism. And then again, in human body, the mechanism, they are not independent. They are depend on other mechanism. Okay. So then you need to study whole things here. If you want to say, okay, this is cancer or inflammation or diabetes, the best one is to also look at all the metabolite, not only one single compounds. Okay, otherwise you will be biased. Like you see the elephant, yeah, you see part, then you will bias, right? Okay, yeah. So metabolomics actually is following the previous omics analogy, which is genomics, so proteomics, especially if you see from the pharmacology part, yeah. So you are actually not need to think about what is the gene expression, what is the effect on the gene expression, yeah. After you have this the environmental factor. So what is the effect to the gene expression? What is the effect after that to the protein profile? And then lastly, to the metabolic profile. Okay. Okay, so actually I already explained this during my first lecture, but actually I need to see deeply, yeah, to be more clear, to be the clearer, yeah, picture how we can apply, yeah, this metabolomics to each of this activity, yeah. Firstly, I would like to, the first approach is we would like the first study is would like to profile the bioactive compounds in the extract, yeah. Which one has been reported, which one is not, which one is known compound, which one is not. And then if it is the compounds already known and also the activity already known, so no need to isolate, right? And then if you want to publish, yeah, the isolated compounds, then you should be sure that what you publish is novel. So meaning novel can be the both the compounds is new, or the activity is new, or one of them is new. Okay, so it means that you need to have the mapping, yeah, mapping of compounds which you think that promising to be isolated. So that's one, yeah. And also maybe you want to study the synergism effect among them is also can be used, yeah, in this for metabolomics, can be used for this goal, yeah. Secondly, actually is the, yeah, after you know the profile of your herbs, and then you also feed, yeah, the animal with your herb extract. So then you need to check their bioactivity and also toxicity, yeah. And then when you compare this toxicity data and bioactivity data, then you will get the therapeutic index, right? So you compare between them, so you can select which fraction or which extract is potential, which has low toxicity and high bioactivity. That's the best, yeah. But to understand what happened in the animal, yeah, what happened to the bioactivity, what happened to the metabolite, yeah. So the best way is to profile all the metabolites, yeah, on the metabolites of the biofluids, yeah, of your animal, right? So the same strategy, yeah, with the study case number 1. Because why? Because now you also apply the metabolomics. When you analyze your data, you need to think about whole metabolite, yeah. Third is if you actually apply the first one, automatically you will go to the number 3 as well. Because why? You can have the calibration regression model, yeah, that can be used for the quality control, yeah, of your herbs, yeah. It means that you are now develop the calibration model in order to predict, yeah, the quality, or predict the quality. In this case can be the bioactivity, or maybe, yeah, it can be used for to know, okay, this plant is from which location, this plant is from another location, or you know already know the grade. For example, okay, this plant is this extract is grade A, this one is grade B. This also can be used, yeah, for these goals, yeah. So we call is the fingerprint, yeah, fingerprint approach. Okay. Okay, this is a fingerprint approach. So you can predict the activity, and it should be very close to your actual activity. Yeah, when you make this model, calibration model, so you make this kind of the model, calibration model. Yeah. So what we want is we want that actual activity and productivity, the regression is more than 0.9. Yeah, meaning this model is valid. Okay. So this model can be used later on to optimize your extract. All right, can be used to optimize your extract. It means that now you don't need to check the bioactivity one by one. Yeah, meaning that after you get the calibration model, what you do is just you analyze the fingerprint using what? Using very simple instrument, which is FTIR. Yeah, because if you check the bioactivity, it is expensive. If you check, for example, enzyme, one IC50 measurement is cost a lot. But if you use only FTIR, it's very cheap. You can get the second. The result can be get the second, right? So the cost for research can be reduced a lot, right? So that's why this quality control methodology, this calibration model is very potential to be used, for example, for the RSM. Yeah, for the RSM, response surface methodology. Yeah, normally you have, for example, different parameter. Yeah, in this case is, for example, the moisture content, the air velocity, the time, for example, how when you dry the extract, for example, right? So then you see what will be the parameter. For example, what is the for example the profile? Yeah, what will be the activity? Yeah, what will be the activity of the product or optimize or the what will be the activity of the extract result produced, yeah, from this parameter. So maybe in RSM you have like 20 parameters that need to be checked. So if you check one by one and then check quality, meaning the bioactivity, it will be costly because you go to enzyme or you go to the cell line, it's very expensive. But if you check them only using FTIR, then nothing, yeah, the cost is much less, yeah. So then you will get the optimized condition for your extract, yeah. Okay, this is actually I need again to show you because this is the most important slide, yeah. So of course I already explained before, yeah, but in order for me to explain in better today, so I need to again review, yeah, this flowchart. Okay, last week, yeah, I explained regarding the how we do the metabolomics approach, yeah, in this research, yeah. So you need to have the plant sample with different quality. I will explain what the meaning of different quality. I will have the data, yeah. So different quality can be, for example, different solvent, different varieties, or different not varieties, they are different collocation, yeah, for example, or different drying condition, or different age, yeah. So any means, yeah, as long as they are different quality, then you can use in this experiment. So just say that you have 5 parameters, 5 different quality. For example, you have different 5 solvent. Yeah, so you have 5 solvent, and each of the quality you need to have replication, 6 replication. So now you have 30 samples. So all these 30 samples, you need to check their chemical profile. Okay, you need to check their chemical profile. How using what? Using the instrumental analysis, using NMR, LCMS, GCMS, and then for fingerprinting, you can use FTIR or TLC. Okay. So for this chemical profile, yeah, should be analyzed. This analysis should be applied to all these 30 samples. Okay, and then at the same time, you also need to check the bioactivity. Yeah, so it can be in vitro. Nowadays I use in vitro, but later on I use in vivo after I develop my zebrafish. Yeah, so okay, just say in vitro. So from this, if you use in vitro, then now you have the data, all this data, then you can combine the data from this in vitro test. Yeah, there is data now. Data from the in vitro test, data from the instrumental analysis. Two set, yeah. Yeah. So these two set of data need to be combined. And analyzed using the statistical method we call as the multivariate data analysis. Yeah, from that, then only we can identify the bioactive compounds. Yeah, and then also from this, then you can also get the calibration model. So this multivariate data analysis actually is kind of the calibration or correlation model based on the multivariables. Yeah, so you make the model based on the multivariables, which is variables coming from the Is metabolite. Yeah. So if you have this calibration model, then you can predict the quality of the plant sample. Okay. And then another thing is the in vivo. Yeah. If you use in vivo, now also interesting part is if you have in vivo, then each of the serum, yeah, or each of the urine, the metabolite in each of the serum or urine can be profiled using the NMR LCMS, for example, right? And in this case, because you have the set of data, yeah, then you can also have the multivariate analysis, and then you can find the biomarkers. If you combine with the data from this profiling, then you can also get the mode of action. Why, for example, quercetin can reduce the glucose from which pathway? Yeah. Or also you can have, if you didn't use this metabolic profile, only use this metabolic profile of your biofluid, you also can explain why, for example, this rat become diabetic, which metabolite affected, which one is suppressed, which one is increased. Yeah, so then you can have some kind of predictive mode of action, right? Why in diabetic, for example, the glucose increase, for example, right? So there must be the connection with the protein or the gene, yeah, or there may correlate with other metabolite as a precursor, for example. Okay, so that's why this is all the pictures, yeah. So whole the application that I already mentioned before actually detailed in this flowchart. Okay. All right. Then why I use the multivariate data analysis in this case, right? So I always in this here I use multivariate data analysis. Another one I also use the multivariate analysis. Yeah. So why? Because I use the multivariables which is produced, yeah, from this instrumental analysis. Yeah, I use this one because of I have these multivariables now. Okay. Okay. For example, here I use NMR. Yeah. If you use NMR, you have so many peak here. You are not talking about pure compounds here, right? Some people said, okay, why you use NMR for the complex metabolite? Normally we use NMR for the pure compounds. Yeah, this is different goal. You can use NMR for illustrative structure of the new compounds, and then I agree. Then you use the that NMR for these pure compounds. if this is new and nobody report before. But now if you know that the compound is very common, for example, like glucose, yeah. So easy to see here, where is it? Right? This is like to try to find here, right? And then you can connect with the 2D NMR, or if you use LSMS, you can use fragmentation MSMS. Easily you can identify the structure from the, if it is known compounds. But if it is the unknown compound, yes, you need to isolate them, yeah. And then later on you go to NMR and you use that for elucidation of the structure. Okay, say I come back again, yeah. Okay, now you have, for example, from the NMR, you get this one. Okay, so now I use variables, yeah. Each of signal peak of this peak actually is bin, yeah, bin area, yeah. Every 0.05 and 0.04 ppm, chemical shift 0.04. Then I bin it, I cut, I slice one by one here. Slice, yeah. Not we are using manually, we use actually the software, right? And then after that we get the peak area. So each of this bin chemical shift becoming one variable, yeah, becoming one variable, which is we call as K, yeah, variable. And then the number of variable we call as K, yeah. So this one can be 100 of variables. So it's not becoming one variable anymore, becoming many variables. Can be 100 variables and even can be 1,000 variables. Okay. Yeah. So then another one is observation. So actually this is coming from this spectrum is coming from one sample, but I may have another samples. So the sample can be 30 sample, 40 sample, depending on our experiment. So the number of observation we call as N. So now we have the number of observation, number of variables, meaning that this is number of samples and this is number of variables representing the metabolites. Okay. So now what we get is number of samples, and here is number of variables representing the metabolite. It can be 100. Number of samples, 30 already a lot actually, right? It can be 1,000. It can be, if you get 100, is already too much, right? So if you use the metabolomics, this is the figure of your data. Okay, you have few observation, but you have too much of the variables. So if you use this, not using the multivar analysis, then you will have the error, especially the type 1 error, false positive, etc. Right? So if you have, for example, the variables, if you have more variables, the risk is will increase. Yeah. So meaning that you need to reduce now the variables. These variables need to be reduced and should be less than number of observation. Okay, so how to do that? Then you use the multivariate analysis in order to get the latent variables. So meaning that if you have 1,000 variables, you need to reduce the variables, yeah, and then the new variables we call as the latent variables, yeah. So this will be the projected variables. One variable, latent variables, now is represent of sum of variables. Yeah. So now, if for example, now before you have 1,000 variables, now you only have 10 variables only. And each of the variables is representing the many of variables previously. Okay. Yeah. So this is like this. Yeah. Projection. You have many variables here. Each of this arrow is representing one variables. Maybe you have 1,000 variables. Now when you project, now you only have become two variables like this. Yeah. So that's actually the idea of the multivariate analysis. We want to use the latent variables. Okay. Okay. Now I think I go forward to the study case number 1. Yeah? So study case number 1 meaning that the approach, yeah, the application, yeah, before I mentioned before, yeah. So actually that one is the profiling of or identification or mapping, yeah, of the metabolite in the extract. So I use okay, I forgot one. Actually there are 3 here: GCMS, NMR, and LSMS. Yeah. So this is coming from 2 publication that we made before, yeah, based on our research. Okay, now you have this plant sample, different quality, and then we have this chemical profile here, yeah, chemical profiling. And the activity test that we use is the in vitro only, yeah, based on the alpha glucosidase inhibition activity, which is related to the diabetic. Okay. Okay, we use the Satria malayana, which is known as the salung or loning, yeah, in North Korea and North Sumatra, is used by the Karo people to treat diabetes. Is also part of the Riau, yeah, or Jambi or the South Sumatra. It also use this one is to treat the diabetic or kind of the some inflammation. Yeah. So based on this, we need to know what is the compound inside. Okay. Okay, so this from this table 1 actually is the IC50, yeah. Okay, from protein inhibition, then we convert to IC50, yeah, and then we get the each of, now we have the five different quality of sample. Third is the methanol 0%, meaning that's in water, 25% of methanol in water, 50%, 75%, 100%. Okay. So now we have different quality of sample in this case, right? We have different quality of sample, and then replication. So replication as well. This is N is 4. So 4 replication each of the sample. Okay. So now you have the C50. Sorry, I have another table. I overlooked this, which is also showing the C50. Yeah. And then they found out that the most active one is somewhere like 0% or 50%, something here. Yeah? Okay, and then another one. So we test the activity of each of the sample. Second wings is we try to analyze the chemical profile from each of the instruments. So we use GCMS. So this is the chromatogram, yeah, of GCMS, and then this is the putative metabolite detected by GCMS, and then this is actually is from the comparing to the NIST database. Okay, so only the one which is similarity index more than 90% we consider, yeah. So we have now 9 compounds from GCMS, and then another one is from the NMR. You see NMR, we have proton NMR, and then we go to the J-resolve. So you see here, for example, in proton NMR, you have the overlapping peak, right? Overlapping peak. So it's difficult for us only to see by proton NMR. So what you need is go to J-resolve, coupling resolve, in order to know, okay, this compound actually is what? Is it multiplet? Is it the triplet or singlet, or triplet or 3 singlet? We don't know. But when we see here, for example, number 10 here, yeah? So actually this is triplet. Okay, and this one, this is doublet or two singlet. So when you see here, this is doublet. Okay, doublet, right? So we also can see when NMR, you can see this is not clear. So but J-resolve, you can see, okay, actually this is coming from this 1, 2, 3, 4, 5, 6. So 6, yeah, sextet, yeah, and another one overlap with 1 singlet here. So actually overlapping somewhere here, right? Yeah, so we not only see from the one dimension, we also can see from two dimension by J resolve. Later on, when we already did the some kind of prediction, then we need to correlate with the SMBC spectrum. Yeah, and you see if there is coupling from proton to the carbon, right? So identification not only simply by comparing the data to the database. Database can give you number of compounds, but how you select the compounds is depending on your knowledge on the NMR, right? So you need to see what is the coupling constant, what is the splitting pattern, what is the chemical shift, what is the relation, what is the coupling to carbon, and then is there a database. So only that you can identify the compounds. Also, you need to also confirm, yeah, the additive compounds by mass. Yeah, so you see the mass. What is the mass, and what is also the fragmentation? Yeah, after the mass MS, is that tally with the UNMR or not? Right. So here actually the myoinositol, yeah, you have the coupling constant, what is multiplicity. Yeah, so from here only you can find that. But also in the discussion actually we mentioned about the SMBC. So SMBC, you can see the coupling between this proton and carbon, and then fit to our putative metabolites. Okay, from LCMS, yeah, from LCMS, we found some of the compound which led to the bioactivity, and some alkaloid, the isophenolic compounds, and also the terpenoid. Yeah. So again, yeah, the database will give you some of the putative compounds. So from 1 Z, yeah, from the parent ion, they will from the parent ion, they will give you some names, yeah. And then of course you have the fragment ions. So from the fragment ions you find, yeah, some of the ions here which is fragmented from the parent ion. So in publication you need to explain one by one, this is coming from what. For example, this is before it's 315, again going to 300, and going to 297, and going to NS, etc. until 27. So what is this? You see, it's not simply compared to database. You need to explain, yeah. Okay, this is coming from what? Okay, so if it is in tally, yeah, then you can give the tentative metabolites. What is this? Okay. Okay, now you already have these two, yeah, data set. One is the bioactivity. Secondly is your metabolic profile from GSMS, LSMS, and also NMR. So now when we go to GSMS, you can have this. This is actually is result from the multivariate data analysis, right? From the result of the multivariate analysis, you will get the score plot and loading plot. Yeah, this is actually projected. Yeah, so you have projection. So now I told you before, we have now the new variables or latent variables. In this case, we have first variable is PLS component 1, and second variable is PLS component 2. Okay, so now you get this score plot, and you see some difference between among the samples. Of course, some of them may be outliers. For example, it's 100. So, but don't worry. Yeah. When they correlate with the bioactivity, then this statistic is valid. Yeah. And then you get the loading plot, and then anything which is close to the Y, this is Y is bioactivity. Yeah. Are the compounds, are the spot which is related to the activity. So whatever close here, yeah, is the one which is active compounds. Yeah, there are actually the the value in order to say, okay, in this region we call as still active. Yeah. For example, another region, for example, somewhere here, is not active anymore. Yeah, this is no relation to activity, but anything which is close to this blue, this is Y, yeah, then it is the interesting spot, which is related, which can relate to the compound which is active. Yeah. So then we found some of them. Yeah. So this is palmitic acid, monopalmitin, tocopherol, epicampestrol, etc. Yeah. So anything which is close to the blue is the one which is active. All right. And then also NMR, the same case. Yeah. So you also have the score plot, you have the loading plot, and anything, any compound which is in the same site of the IgI activity, meaning that compound is the one correlate to the activity, meaning positive correlation to the bioactivity. Yeah, in this case you have compound 10 and compound 11. Yeah, so we already know this 10, 11. Last time is this one. This is the hydroxyphenyl pyruvic acid. Another one is the glutamine. Other one like inositol or sucrose or glucose is no relation to the activity. Okay. Yeah. So because their error bar here is too big, so there is no relation. Yeah. Okay. So what about SMS cutoff? The same things. Yeah. So we interested only the compound which is in same side, yeah, of the blue one. This is the bacterial compounds. You see now we already have make the mapping, right? Like this one. We already have the mapping. See, mapping, right? So anything which is close here is the one which is active. And this compound may be not reported before. Yeah, because when we check in the database, it's not matching. Yeah, it can be maybe they have still overlap with other compounds, or maybe this is really non-reported compounds, which is it can be new. So when you want to isolate, so please consider the one which is not cannot identify here. Okay, as well as SMS as well. Right? Okay, so now when you go here, yeah, the last time we found the palmitic acid is the one which is active. Yeah, so when we check, yeah, you buy the standard and you found, okay, this one is active. Yeah, is in line or tell you with our metabolics finding. But then when we try monopalmitin and tocopherol, it's not active. It's not active. So why they're not active? So this is what if you go to the Bio Assay Approximation, this is what you will get. This monopalmitin is not active. Also tocopherol not active. But metabolomics say this is active, the one which is involved of the activity. So why they're not active? Because actually there is synergism effect of this monopalmitin or alpha tocopherol with other compounds. So what we did actually is spiking. Yeah? So spiking of this monopalmitin to the extract. So whatever add monopalmitin, for example, here is you add 0. In here no adding, and then you add 0.35 microgram per liter, 0.5, 1, 2, 4, and you find that there is increasing activity. Okay, meaning that there is synergistic effect of monopalmitin with others. Yeah, so we found that metabolomics can explain that monopalmitin is the one which is active when they are in the extract, and when we try, yes, it's true. As well as alpha tocopherol, when you check individually, it's not active. But when they are in the extract, yes, the tocopherol giving activity, you see the increasing. Yeah? of the activity here. Okay. Yeah, of course you can have this docking to understand the molecular level. Yeah, I think I don't need to explain. This is like how we explain, how we get the fragmentation of SMS. This is how we explain. So you need to know the sum of the knowledge on the SMS. Yeah, so that one is about the study case number 1. Dr. Ena, should we continue or we have kind of QA session now?
Maybe there is continue to case number 2, and then we take the questions after you finish.
Oh, okay. So if you have questions, you just raise your hand, yeah. So maybe you don't understand, so then just raise your hand, right? Maybe this is quite new for you, right?
Yeah, thank you.
Okay. For the application number 2, yeah. For the application number 2, actually we try to understand what happened on our animal models, yeah. So in this case, I use rat and zebrafish, just compare it. Actually, nowadays, I really interested on zebrafish, yeah, because why? Because I want to use this model for my metabolomics work to understand, to explain the mode of action of each of the bioactive compounds in herbs, right? Okay, before I find the zebrafish, I use rat. Yeah, I use rat. So, okay, last time you used the Carantia, Mamordica Carantia, pari. Sini paria. So you see here, if you use only one glucose, one parameter, which is glucose, yeah, what you find out that, okay, this Mamordica can normalize the blood glucose level, right? So this is what happened. Okay, metformin as well. Yeah. So actually, what we get here is try to mimic, try to get this induced diabetic rat. Yeah. So we make it obese, and then after that we inject a little bit of STZ, yeah, streptozotocin, yeah. So and then you see the glucose level is now is 4 times higher than normal, right? Normal is still like this, and then all of this one is after injection, then become glucose level is very high. But after treatment, you see that metformin and then also treated by the Mamordica going to normal, right? The same as the normal. But the diabetic one still high. Okay, so but from here is if you only consider only one metabolite, then our conclusion that Mamordica able to normalize the glucose level, right? Okay, but however, yeah, when you apply the metabolomics, you see different conclusion. What is different conclusion? Yeah, because now in metabolomics, you are not dealing only with the glucose. You are dealing with other compounds as well. Yeah, in metabolomics, we try to analyze as much as possible, yeah, the metabolite. Yeah. So you see here, for example, this one is diabetic. And then the normal one is this blue one. So there are discrimination among them, both in plus DE component 1 and another one is plus DE component 5. So both using both variables, they are different. Okay. So when you use metformin as the positive control, yeah, from this table, actually, this is normalized, right? But what happened here? The metformin cannot normalize. The metabolites, meaning the all metabolites, cannot normalize if you consider all metabolites. They cannot normalize. They only can shift, yeah, based on the PLSDA component 5, but not PLSDA component 1. Okay, this is the serum. So meaning that now you have different conclusion. Metformin cannot normalize the profile of metabolites. Only can normalize the glucose, right? What about this paria? Yeah, they can shift, but even better than metformin. Yeah. However, there is still not fully, the profile is not fully normalized because there is some gap here. You see some discrimination. If you see the more closer enlargement of this, you can see they are different. Okay. So now our conclusion is different from before, right? It's not like this. Now we found it's not normalized, right? Okay. What about the urine? The urine is also the same thing. Yeah. So you have, for example, here this STZ. This is the diabetic. Another one is the normal here. Okay. So actually, the metformin and Momodica can normalize only, can shift, yeah, based on the PLSDA component 1, but not, sorry. Okay, this is the normal, yeah, but not based on the PLSDA component 4. So it means that there is still work to be done in order to shift our sample to be exactly the same as normal. So now the claim that the Momodica or metformin can, yeah, normalize the glucose, yes, true, but not the whole profile of the blood or urine. Okay, so actually there is still a lot of work to be done in order to achieve the goal, which is the all metabolites should be normal, right? Yeah, this is actually this how we identify the compounds in the same way how we analyze the compounds from the herbals. Yeah, so you have, this is protein NMR, you go to G-resolve, you go to D, and you go to mass, and then you find all these names. And this name is not simply just compared to the database. No, yeah, it's wrong. You need to check your 2D NMR and also to check by mass. Okay. So now what you get is, yeah, it's complex, right? This is like complex things in the urine or in our biofluids. So not consider only the glucose, but other things here. Okay. Now we try to make this table to simplify what compounds actually is normalized, what compound is not normalized, right? So whenever I say this arrow, meaning that this can be up or down or can be reduced, yeah, can be go down, meaning that if that is up, meaning that if you compare the metabolite after treatment or under this condition, then compare to the healthy one. So when you compare to the healthy one, then you find this is the amount is up or going down. Okay. So for example, this one, the one which is with the already inject with the STZ under obese condition. Yeah. So what we found is that the metabolite hydroxybutyrate higher compared to the normal. This is compared to the normal. But when you treat with the Momodica, it can be normalized. So when you compare to the normal, it can be normalized. Okay. Valine, yeah, here also higher amount, but also in Momodica, it cannot be normalized. So Momodica cannot normalize the valine profile. Okay. Ethanol also high, yeah, compared to the one which is normal. Also Momodica cannot normalize it. So etc. etc. So what this is we found, yeah. So Momodica cannot normalize lactate, cannot normalize ethanol, not valine, but can normalize the hydroxybutyrate. Acetate can be normalized. Glutamate, yes, yeah, cannot be is going to be normalized. Glutamine cannot. Succinate, yes. Creatine, yes. Choline, yeah, in the STZ is normal, but when Mamodica, now we found they are become worse, yeah. Glucose, betaine, yeah, normalized. Glucose normalized. Taurine normalized. Okay, so this is the when you see summarize it, this is what we get, yeah. So actually, this Mamodica, of course, not can also normalize some of the metabolites, but not fully, yeah. So still there's some of the compounds, right? Also the from here as well, yeah, from the urine, yeah, from the urine is much better. Succinate is normalized, oxalate normalized, citrate not, creatine, yes. So you can see the better image, yeah, and the better better shift, yeah, when you check the urine, yeah. So now you can see more, right? But actually, in metabolite here, we're not targeting anything. We just see whole things, which is as much as possible we can. So I use only NMR. At the time, I don't have LSMS. Now we have LSMS. Then at LSMS, we can see more description on this, what metabolite is shifted, which one is normalized, which one is not. Okay. Yeah. So that thing, yeah. So when you use red, but now, yeah, if you see this samples, yeah, actually that one is our experiment using this red. Actually, is only our exercise. Yeah? So, but actually, what we want is this. So, bioactivity test in these wings, you use not in vitro anymore. You use in vivo directly. So, when you use in vivo, you need to have. You cannot use red. Yeah, because imagine you have 30 sample, and then you need to check one by one using red. So how many red you will be killed? And this is ethically not approved. Yeah. So you have, for example, one sample. Even if you have one sample, you need to have at least 200 red. Yeah, 200 red or 150 already minimum, or 100. Yeah, 100 to 200. So if you have 30 sample, meaning that 30 multiplied by 200, so 6,000 red should be killed. Then you are out of business, right? Yeah. So how can we find the in vivo test which is credible? Yeah, but simple. Yeah. So last time when I study in Holland, I see many people using zebrafish. So they try to use the zebrafish for the Alzheimer, dementia, and also some of cancer research. So I come back to my old times. So then I try to see the review from review. Okay, this zebrafish is promising, right? Because this is the one which is good to have for them this metabolomics work. Yeah. Because why? Then you only use 96 well. You put the embryo and larvae there. Yeah. Like check the 96 well using enzyme. Yeah. So each of the well will be containing one larvae or embryo. Yeah. So that one well considered as in vivo test, right? Not in vitro anymore. Yeah. So by this way, then it's possible for us to do in vivo, right? Okay, I try come back long time ago with my master student, Benchola et al. You can read the paper. Yeah. So we use this is larvae, yeah, larvae. So you can see all the LSMS, yeah, because we cannot use the blood, yeah, at the time. So we just try to crush everything. So see, there is LSMS there, right? But anyway, yeah. Anyway, before we go to the larvae, we need to check by adults. How is it? Is it possible or not using the adult zebrafish? So the same same picture. Yeah, if you use the glucose, you found that the plant's herb extract can normalize the blood, can normalize the blood of the zebrafish. But when you go to the metabolite profile, meaning that everything is calculated, everything is analyzed, then you find out that these herbs cannot normalize the blood profile. Same thing, right? So meaning that, okay, then zebrafish is actually is also promising. Yeah. So, but you know, this one is adult zebrafish. Yeah. So what about if you use the embryo, yeah, or larvae? Yeah. So we cannot just withdraw the blood. We cannot, yeah, because it's too small. So what we do is check the gene suppression. Yeah, gene suppression. So actually, this one is going on, yeah, in my lab. If this one is okay, then we shift, yeah, from adult to larvae stage. So in this case, easy for us to get a ethical approval, yeah, because then also the homogeneity, also the cost is also not much, yeah, because we use only larvae. You can breed the zebrafish on one night. You can get 2,000 egg easily, right? So this is I think a feature of our in vivo. Also, we also use for toxicity. I think I will not display here. We already try with toxicity. We find also a promising result and already published in the Pharmaceutics, yeah, Pharmaceuticals, and also toxicology reports. So we follow the OECD guideline for this, and then they agree on this. So we have confidence now to check the toxicity before we use rat, yeah, or mice. Yeah, we use mice to check the toxicity, acute toxicity. For example, now just use the zebrafish. Yeah, zebrafish homology is around 76, I'm not mistakenly, 76. And also the homology of the gene compared to the human is around 86. Yeah. So it's okay. Yeah, you can use zebrafish. So that's why now, if we have all these tools, if we use all these tools, so our problem, our query on the standardization, on how to study this scientific proof, yeah, that's what I already explained before, yeah, can be achieved, yeah, can be the problem because of what analytical tools, right? So then this is open our the possibility for us to do that, yeah. So this is the conventional approach, yeah. Conventional approach is, yeah, in vitro test for the screening, right? Screening in vitro test. One by one, glucose uptake or DPP4, alpha glucosidase, alpha amylases for the diabetic. And then you try if this is active, then you go to the rat or mice. And then you check also toxicity in vivo. And after that, you go to the clinical trial. That's the things, right? In order for you to get the approval, the approved drugs. But this one surely takes very long time. Yeah. Because it's also very expensive. Yeah, you many times you find out in vitro test active, in vivo not active, or vice versa. In vitro not active, but we go to in vivo is active. So this is confusing, right? And also just like gambling as well, right? So what we want is in vivo. The final say is by in vivo. So why don't we screen from in vivo? Of course, we cannot use rat, we use zebrafish. This is in vivo. Okay, if zebrafish said, okay, active, this is active. Because zebrafish homology and also rat is, yeah, almost, yeah, compared to humans, almost similar. Yeah. So when this one already said is positive, then we want to know which mechanism. So mechanism, only you confirmed by in vitro. So in vitro is not using for screening anymore now, but in vitro only to confirm. Okay, to confirm what our hypothesis here. Yeah, so we confirm this one. And after that, we also can do screening not only the bioactivity but also toxicity at the same time. Yeah, here not only the bioactivity but also toxicity. So what you want to see is the therapeutic index is high. So meaning they have the less toxic but high activity. So all the screening is by in vivo, right? Okay, and then we can, after that, we can, if you want to go to testing rat, it's also fine, but also you can go directly to clinical trial. Yeah. So it means that we can save a lot of time here. We can save a lot of time. Yeah. Because our consider now is herbal extract, you know, it's not the pure compounds, it's herbal extract or optimized extract or mixtures. So we will have plenty easily for us to get the samples or extract or drugs. Before we only use the individual compounds. Yeah, if you go to rats, can you imagine how much? If you have like, they need like grams. Now you are talking about it isolate only 10 milligram is very difficult. Now they want a gram, so difficult, right? So but this one, no, we are dealing with something else, right? We are dealing with the complex, and we know, and we have the screening, the active. Okay, so hopefully this will open our what's the future to study to how to optimize the one for the nutraceuticals, right? That's what I would dream. Yeah. Okay. Then also we can also combine the data of these medicinal herbs metabolite with the here the right one. It can be the result of the metabolite. So if you go to the multivar analysis, it become X variable, this is become Y variable, then they can explain, you can connect each other, you can connect and see how they connect, what is their connections. Okay, so for example, in diabetic, for example, which metabolites is affected, so you go there. Okay, which one is not affected, you go there here. Yeah, and then you see their relation with the metabolite. So each metabolite can explain. So it's become, if you use the multivar analysis partially square. So now you can correlate them. Yeah. So this is very good for the molecular level study. Yeah. So in order for us to see in detail what happened. So what if we want to, if our dreams that these nutraceuticals can be our drugs in the drugstore, all these ones should be explained. Otherwise, cannot. Yeah. So again, this one we need metabolomics, and possibly we have used proteomics, genomics to see the relation between gene, protein, and also the metabolite. Okay. So that's the thing is about the study now. Case number 2. Study case number 3 actually is the analytical tools for inequality control and process optimization of herbal product based on the fingerprint. So this is I already explained last week actually. So you have if you have if you have the extract, yeah, even if the single compounds different, you will get different spectrum of what they are. Even if you have trans isomers, they will also different. So some said, okay, there's overlapping. Yes, overlapping, no problem. So we don't want to elucidate the structure the compounds. We want the fingerprint only, right? So if there is some different amounts of compounds there will be difference. So it's fine. Yeah. So of course we will not see this by eyes, but we will our software will count for us. Okay. So again, the same thing, different quality of sample will be analyzed with the different analytical instrument, with FTIR, and also will collect it with activity. So then, yeah, what we want is this. This model. So this model, the R square should be more than 0.9. Yeah. So meaning that if you have the new set of data, new set of FTR data, now you can predict. You just put in the software, yeah, and then they will say, okay, the activity of your sample will be this number. Can we believe on that? Yes, of course, because this is based on this model. Yeah. And a lot of questions can be also used this one for the herbs, which is from another province, from another. No, cannot. But for the in-house quality control, yes. Meaning that you have already set all the parameters, set all the sample condition in a certain range. Yeah. Okay. So, but it is very good for us to control the quality and also study on the optimization of the production parameter. Yeah. So, for example, drying. What is the velocity of air? What is the temperature, etc. Then easy for us to predict the activity based only for the simple FTR spectrum. Okay, FTR, you see that now you have the ATR attached to the FTR. So what you do is just you grind, you grind your you dry the sample and then you grind it, and then you just put in ATR. What happened? Then you can see the spectrum. This one actually is from this grinded powder, easily for us to see. Yeah. So for example, you have, for example, now gaharu, right? Gaharu, people difficult to grading. Yeah, this is like mafia, right? Some said, okay, this is grade A, grade B, grade C. There is no actually the consensus for the grade A, grade B. They just say, there's social people. I mean, this market will say, okay, this is grade A, grade B. If you go to another seller, oh no, this is grade C. Can you imagine? Yeah, but if you have this one, this model, you cannot cheat me. This one is grade A. This one is grade B. This is grade C. Like this, right? Or for example, like vanilla from this location. Okay, they say that, okay, the location from this area is very expensive. Okay, but how you trace that this one is from the area? How you know this comes from the area? Yeah, from example, in our area we have the ginger. Ginger from Kara is expensive. Yeah, others not. So if someone come to us and then, okay, I sell this ginger, and this is the grade from Kara, how we know, right? Okay, because of this document, how we believe. So in order for that, then if you have this FTR spectrum, you put in your software, you know, okay, you are cheat me. This is not from Kara. This is from other location. Okay. So based on what? From the spectrum. Yeah. If they said, okay, this is match, then yes, we call it as fingerprint, right? So there's another things also can be used for application of this one. Yeah. So again, I repeat, this one is not only by this R square and 0.9, but also validation here. I think no need to discuss here, but there is some validation method in order to say, okay, this validation model can be accepted or not. Yeah, there is not like just simple make it and then you can use it. No, there is a lot of validation process on this. All right. Yeah, this is what I told you. Yeah, before is now we can have the mapping and see which spot can be a novel and maybe potential to be isolated. Yeah. Okay. Then our method now to isolate the compound now is not bioassay guided fractionation anymore. Yeah, I read here. Now we want to use the new approach we call as metabolomics guided fractionation. Why? based on this map. So we just isolate them based on that map, but not Bio Assay, because Bio Assay meaning that you will miss the activity when you fractionate, then you change the plan. Then you waste 6 months or 1 year, and then you change the plans. This is a hectic work, right? So if you go to metabolomics definition, you are sure about what is the peak that you want to isolate, what is the mass you want to isolate. So easy. After you get the compounds, then you can have a structure. If you are not okay, maybe this is not active, for example, and then you can spike, okay, to the plant extract. Okay, for conclusion, so metabolomics open the possibility to fasten the identification of novel bio and chemical markers. It can be used for the validation. A validated representation model can be used to predict the activity of new set of sample that can be used for the quality control and also optimization of the production parameters. And then next is we can also see the commercial study on the mode of action. So we can see the new biomarkers or how these metabolites can inhibit enzyme, or how can we correlate between this metabolite in the herbs and also the mode of action in the animal study. And next is the toxicity. Toxicity, normally we get LD50, right? So LD50 is already die, right? But what happened before? What happened before die? What metabolite are shifted? What metabolite affected? So this is need to be addressed. Otherwise, we just said, okay, LD50 this, and what we said, we can see the behavioral study. We can check, for example, the teratogenicity study. You check maybe the deformation of the brain, deformation of the heart. But actually, before the deformation of physical of the organ, there is something happen in the metabolite. There is something happen in protein level. So if you have this omics, we can also study this toxicity. We don't want that when we have these herbs, then actually there is toxic there. Some toxic effect already working on that early, before the LD50. So hopefully, this can open our way to have standardized with a clear picture of this dose response as also toxicity. And then if we have this one, hopefully it can be used for the treatment of disease. So I think that's all, moderator. From me, maybe there is a question and answer. Question and session, QA session now.
Thank you, Professor Alfie, for the talk. That was very interesting talk and surely has sparked some questions like we see in the chat box. I would like to remind all the attendees that you can raise your hand if you have any question, or you can also type it in the chat box. Questions in both English and in Bahasa Indonesia are welcome, and we'll try to translate your question as best as I can. Now we have question from Ibu Dinar. Thank you for the comprehensive presentation, Professor Alfie. Let me ask you a question related to the different quality of the sample. We are common to have here in Indonesia one of several trees which have empirically to have a biological activity. Is it not possible to have it into metabolomic study because it has no different varieties? If it is possible, how can we do the metabolomic study in this case? Thank you for the answer. Prof. Alfie, if you please answer the question.
Okay, thank you, Dr. Dinar, for your questions. Of course, if you don't have different varieties, no problem. We can still study on that. So, for example, you can see the different quality on different site. For example, the drying effect or solvent, solvent different, and also using extraction, and also maybe you have also the different age, or you have different storing condition, or you have different drying condition, or many things that make the sample different in their quality. So the quality not only due to the variety, but also many parameters that they are different. So as long as they are the quality is different, yeah, then we can see, yeah, the different of the bioactivity, different of the meta profile, and then we use the MDA, yeah, multivar data analysis, to calculate that difference. Yeah. So no worries. Yeah. If you don't have different varieties, then it's fine. No problem. If you have a different location, it's also fine. Okay, Dr. Rina, is that answer your questions?
Yes, thank you, Prof. Alfi, for the answer.
Ibu Dina, do you want to follow up the question further?
No, thank you. Yes, it is enough. Thank you.
Thank you, Ibu Dina. So from what we can conclude, then we can actually still do the metabolomic study, although there is some little bit variety or variance in the quality, like the source of the material. Is that correct, Prof. Alfi?
Yes, possible. Problem.
Yeah, so that should be no problem in doing metabolomic study for herbal medicines in Indonesia, given that we may have different qualities from the supplier.
Yeah.
Is it just like that? Okay.
Yeah, as long as you then you should know also the history, yeah, from the plants, not only okay the supplier 1 A B C, you need to ask this is from where, and then they have some mixture, maybe there has a mixture of plant, then difficult. If you buy from the different farm, then it's fine.
Okay, thank you very much, and I also would like to thank you for being one of the pioneers in the study, given that we have huge varieties of herbal medicine and that someone has to start to make the big metabolomics database. Thank you for your research.
Yeah.
Is there any other questions from other attendees? Maybe from the students?
Mungkin dari saya dulu.
Oke, Dr. Sabtono.
Yeah.
Oh yeah.
Mohon maaf, ini saya pakai bahasa Indonesia ini, Prof. Alfi.
Ya, saya juga bisa bahasa Indonesia.
Ya, pertama saya ucapkan sekali lagi terima kasih pada Prof. Alfi atas sharing pengetahuannya pada kami, dosen dan juga mahasiswa kami. Tadi bisa disampaikan oleh Prof. Alfi terkait aplikasi metabolomics yang luas ya, selain untuk herbal drugs juga mungkin tadi juga nutraceutical ya, namanya itu juga ternyata juga berkaitan. Nah ini mungkin untuk benchmarking ini Prof. Alfi ini, untuk di kurikulum kita ini juga sudah mulai mengenalkan ya untuk pendekatan metabolik ke mahasiswa kita, termasuk juga nutraceutical tadi. Tapi memang untuk kedua topik tersebut, metabolomics dan nutraceutical, ini baru masuk di mata kuliah pilihan untuk mahasiswa kita yang di tingkat advance seperti master advance. Mungkin untuk perbandingan untuk di kurikulum di sendiri seperti apa, Prof. Alfi, untuk
ya itu
dan diberikan di tingkat bachelor dan
ya, kita untuk di sini kan itu sebagai elektif ya, tips tadi, tapi dia harus lulus dulu di farmakognosi satu lagi Pharmaceutics Analysis ya, Pharmaceutics Analysis 1 dan 2 harus lulus, kemudian farmakognosi harus lulus, kemudian Herbal Science lulus. Nah, setelah itu baru dia masuk elektif biasanya untuk sebelum dia masuk ke lab. Nah, jadi sangat tingkat akhir.
Jadi relatif similar sih, Prof. Alfi. Untuk kurikulum kami juga memang ini Prof. Jadi sifatnya general, kita belum memisahkan antara pharmaceutical science dan klinik. Jadi semuanya diberikan, termasuk juga yang tadi elektif untuk metabolomi dan nutrisi. Baik. Mungkin yang kedua nih Prof. Alfi. Tadi juga Prof. Alfi juga memberikan contoh-contoh spesies yang sudah dikembangkan, termasuk juga tadi untuk antidiabetik tadi. Mungkin juga mungkin Prof. Afi juga bisa memberikan share untuk pengalaman di Malaysia ini seperti apa, apa namanya untuk tingkat komersialisasi, apakah juga sudah mulai apa ya masuk ke dalam hilirisasi untuk produk-produk, misalnya tadi untuk yang pare tadi ya. Pare saya kira juga di Indonesia juga salah satu apa ya yang banyak ditemui di menu sehari-hari untuk Prof. Awi ya.
Ya.
Pare gitu. Mungkin bisa share pengalaman di Malaysia, Prof.
Ya, makanya sekarang ini kan sebenarnya masih tingkat nutraceuticals kan, ya, belum belum bisa ke drugstore itu. Artinya kalau nutraceutical itu kan dia tahapannya kan kayak suplemen aja gitu ya. Jadi belum apa namanya masih panjang ya pergi ke. Tadi kan udah saya lihat kan, kalau saya pakai Metabolomics ternyata dia punya profil darah itu nggak balik ya ke normal. Sedangkan kita mau dia sebagian balik ke normal gitu. Kalau kita menggunakan hanya glucose level, apa bedanya dengan yang sudah ada kan? Metformin udah aja metformin aja gitu kan. Yang kita mau kan dia memang balik semua profil itu memang tugas yang berat gitu. Tapi secara logika ya, memang sepatutnya kalau kita mau men-treat penyakit dalam tubuh kita, memang kita harus pakai yang kompleks. Karena apa? Karena mekanisme dalam tubuh itu kan kompleks. Kita kira ada 1000 metabolit, tapi mekanismenya bisa jutaan atau mungkin miliunan, mungkin triliunan mekanisme. Mustahil bagi kita untuk melihat satu persatu mekanisme aja mungkin satu PhD study ya. Ini ada jutaan. Wah, itu belum lagi koneksi dia dengan satu mekanisme lain. Jadi, yang paling bagus itu adalah kita lihat secara keseluruhan aja, ya, seluruh metabolit, seluruh protein, seluruh gen. Kalau metabolomics masih agak mudah karena metabolitnya kan paling 1000. Nah, tapi kalau gen, belum stabilitasnya, belum proteinnya. Tapi jangan khawatir, kalau kita nggak pergi ke gen genomik atau proteinmik, paling tidak ke metabolomics saja dia udah normal. Kalau metabolitnya udah normal, dipastikan protein profil dengan genom profilnya juga normal. Karena itu adalah hasil akhir. Ya kan? Jadi jangan khawatir dengan kesusahan yang ada di genomik atau proteinmik. Kita fokus ke metabolimik aja udah bisa menjawab ya. Jadi yang terjadi di Malaysia, saya nggak berani gitu mengatakan oke lah, saya punya glucose level ini, saya nggak dengan mudah mengatakan oke lah saya buat ini untuk saya jual gitu, itu hanya suplemen aja. Tapi kalau untuk drugstore itu saya terus terang saya pengen ngelihat bahwa itu memang profil darahnya itu kembali normal seperti itu. Kalau enggak ya udahlah pakai aja metformin kan udah sama kan, glucose levelnya juga berkurang normal kan. Udah itu satu lagi toxicity ya, toxicity study-nya juga kan ternyata sebelum LD50 kita dapat sekian contohnya LD50-nya 500 mikrogram per mililiter contoh ya. Nah, tapi sebelum itu sudah terjadi sesuatu di sana, 100 mikrogram semacam kan. Apakah itu membahayakan atau tidak? Nah, itu juga harus dikaji. Jadi di Malaysia sendiri tahapnya masih kayak nutraceuticals, enggak segampang itu. Itulah sebabnya kenapa kita mencari semua tools-tools ini. Kita harapkan dengan tools ini akan lebih mempercepat ya, menjadikan herbal ekstrak ini sebagai obat ya, kayak terapeutik aja, terapi ya. Tapi itu banyak kendalanya yang jelas bahwa kita berhubungan dengan metabolit yang banyak di dalam ekstrak tadi, tidak hanya satu ya. Dan bagaimana belum lagi stability dia kan. Nanti setelah ini stability lagi ya, stability study dia, formulation dia gimana, so masih panjang ceritanya. Tapi paling tidak kita sudah memulai gitu ya dengan cara ini. Nah, terus juga satu lagi, kalau kita berbicara tentang herbal, kita tidak bisa lepas daripada proses semua ya, optimasi di mana dia paling optimum ya kondisinya. Dan satu lagi kita juga nggak mau bahwa nantinya herbal ini cuman jadinya seperti random aja kita ceknya. Kita harus ngikut traditional use. Ya, kalau random nanti yang terjadi ya kita seolah-olah trial and error. Kalau kita ambil daripada traditional use, contohnya seperti jamu. Jamu itu adalah contoh yang paling bagus ya. Jamu kita jangan sampai hilang itu ilmu jamu ya. Saya baru berbicara tentang satu herbal ya. Kalau di jamu kan banyak campur-campurannya kan. Ada campuran turmeriknya, ada campuran dan lain-lain. Nah, campuran-campuran itu harus kita study ya, harus kita cek lagi. Oke, tanaman ini ternyata untuk mengurangi toxicity di hati atau di ginjal. Itu belum sampai sana itu. Ya. Jadi sangat menarik ya. Hanya kita bisa lakukan dengan metabolomic kemudian. Saya terus terang aja nih sedang mengumpulkan formula-formula ya untuk dites ya dengan kita punya zebrafish nih. Nanti kita lihat kira-kira formula mana yang betul-betul bagus gitu. Nanti dari formula yang ada itu kemudian kita optimasi. Nah, ya jadi memang lambat tapi ya gimana ya? Kesanalah daripada kita buat yang tadi isolasi, wah itu tambah lama lagi gitu. Ya tambah lama lagi ya. Begitu mungkin Pak Dr. Sabtono ada bisa ya.
Baik, terima kasih Prof.
Ya.
Thank you, Pak Sabtono, for your question. We actually have one more question from Ibu Dinar in the chat box. I'll just read it out for you, Prof. Alfie. Regarding the metabolomic guided fractionation, what are the obstacles during the study? As your explanation say that it seems we need to have multiple disciplines to do that.
Yeah, actually this one, yeah, if you are dealing with this isolation, I'm talking about purification in this case. So in conventional approach, we use the Bio Assay guided fractionation. Yeah. So now what we try to apply is instead of we check the fraction, we check the bioactivity on each fraction. Now we are not dealing with that. Now before we are fractionate, we will have kind of map of the extract. So which compound or which peak is should be isolated. Yeah. So in this case, that's why we call it metabolomics guided fractionation, because we are based on the metabolomics result. Yeah, the fractionation is based on metabolomics result. So what is the obstacle? Yeah, I think this one even more simpler. Yeah, because why? Because now after fractionation, you don't need to check each of the fraction. If you have 20 fraction, or now you need to check one by one the activity. Now what you can do is just check the LSMS. I think if I have a choice, you need to check the activity or need to check the GCML LSMS. I prefer the LSMS because easy, right? So, but it will become complex when you are talking about, for example, you want to relate, between this herbal metabolite versus the metabolite in our body, in animal or animal, So then we need to have a multiple discipline, of course, with the biochemists and then with pharmacologists, and then see how they are correct each other, right, between the metabolite in our body and metabolite in the samples. So but actually this is the capacity is there, but for the fractionation itself, metabolic fractionation itself, I don't think this is become more complex. Is it for me even become simpler, because now after fractionation, and now I know, okay, this peak what I target. So even I can directly, directly going to other fraction. I don't need to check. I don't care about others, other fraction. I don't know. There is nothing there. See, quite fast. Yeah. Okay, maybe you can answer your question, Dr. Dinar.
Ibu Dinar,
Yes,
do you have any
Prof.
further comments?
Do you hear me?
Yes, yes.
Yes. Answer, Prof. Alfi. It needs a comprehensive study to do this metabolomic, and respond to your statement that now you are doing, you are collecting herbal formulation to do this study. Pak Sabtono also have several herbal formulation, Prof. Alfi. So Pak Sab may collaborate with you. We have several herbal formulation, and it is already commercially sell. And I think the scientific information behind this needs to be investigated mainly with your metabolic study. It will give a comprehensive result. Yeah.
Yes. So you can collaborate. You can, for example, send your FYP student here. Yeah, you can send. We have actually kind of the scheme here. Yeah, we have MOU with Brawijaya University and Erlangga. Brawijaya, every year they send like 11 students coming to our lab. So all chemical is from us. But the living cost we are not provide. So they come for example 3 months, 3 months, and then they will work whole day, and then at least excluding the weekend, of course. But then we get totally satisfied with the student, because when people, especially from Indonesia, coming, we like it, because normally the result is good. So then we can publish together on this, and also if you don't want to publish, we can go patent, no problem. Yeah, you can also have this agreement before, so this results will be patented or published. So as long as you can provide the student, FYP student, is fine. Yeah? You can try next year, for example, but should be bear in your mind. But the visa application is 6 months process. Yeah. So for example, in the next year, normally we open for in September intake. Yeah, September intake. So by March, the students should be apply for the visa. Yeah. So at least by March should they apply by visa. So we get visa maybe in April. Maybe in May or June. After that, they can plan the study. You see. So I think that's one of the possible scenario for us. Yeah. I think we already have the MOU between our institute, right?
Yes, we still have
MOU, right?
I think so. But we should pay attention for the period. I'm not sure it is already expired or not, but we can extend it, right?
Yeah, but we can extend it, and then if possible, actually in our university, if we can make the MOA saying that maybe if there is a student from you, maybe for the dormitory is free, yes, possible. We can make the MOA, so it can reduce also the cost for the dormitory, for example, right? So it's also help for the student, right? So the chemical is coming from us, and then also the dormitory also free, for example. Yeah. So then for us is why then we have the free. We don't need to pay the living cost of student, right?
Yes, for your information, we have here also the international scholarship for other graduate students as well. So
Yeah. Asian
probably we
student.
can, yes. So
Asian
we
student.
can use this because it needs agreement before with the international university, and if we agree, then we can proceed with this. Yeah, Pak Sab.
Yeah, we can start already the process. Yeah.
Mungkin saya menambahkan sedikit informasi, Prof. Alfi. Jadi memang tadi sedikit Bu Dinar memang benar mengingatkan tadi, memang MOU kelihatannya berakhir tahun ini. Jadi mungkin kami nanti akan juga menindaklanjuti untuk perpanjangan MOU, dan tadi juga mungkin bisa menambahkan turunan MOE terkait tadi tawaran Prof. Alfi. Terima kasih untuk tadi kerjasama riset tadi. Yang selama ini kita lakukan yang rutin Prof. Alfi, pengiriman untuk e-cop itu kita beberapa kali sudah mengirim mahasiswa kita. Tapi kemarin terhenti karena COVID. Jadi ini terhenti. Mungkin dengan membaiknya situasi, mungkin tahun depan bisa kita inisiasi kembali untuk e-cop, dan juga tadi ditambah tawaran dari Prof. Alfi mungkin dengan skema tadi yang terkait riset. Terima kasih.
Iya, sama.
I think there's no more question. So I would like to thank you again, Prof. Alfi, for being here with us, sharing your knowledge and expertise in metabolomic study of herbal medicines. And with that, we have concluded the first part of today's webinar, and promptly we will start the second part. The second part is the 3D printing and emerging technology in pharmaceutics by Assistant Professor Dr. Awis Sukarni bin Muhammad Sabri. If I could get the spotlight to Dr. Awis, please. Thank you. Okay. Good morning, Dr. Awis.
Good morning.
Thank you for joining us in our webinar series. Soon will be your talk in 3D printing and emerging technology in pharmaceutics. I would like to read out a little bit of your profile before we start. Is that okay?
Yeah, okay, thank you.
Dr. Awis Sabri is a graduate from University Malaysia Terengganu, or UMT, in 2008, in Bachelor of Science with honors in Analytical and Environmental Chemistry. He then pursued his master's degree in chemistry from University Kebangsaan Malaysia in 2012 with UKM Zamala Scheme Scholarship. Afterwards, he continued his study by pursuing a doctoral degree at School of Pharmacy, University College London, or UCL, sponsored by the highly competitive SLA scholarship. Dr. Awis' research interest is about pharmaceutical formulation and analytical chemistry. With that, his primary focus of research is on optimizing drug delivery and 3D printing. Up until now, he has several articles published in high-impact journals and has won several awards in innovative competitions. Dr. Awis is a registered chemist and the current head of Pharmaceutical Chemistry Department at Kuliah of Pharmacy, International Islamic University Malaysia, IIUM, the same university as Prof. Alfi, as well as the chairman of Institute Kimia Malaysia at Pahang branch. Now, without further ado, Dr. Awis, the floor is yours.
Thank you very much, Dr. Erna. Let me share my screen. Okay. Okay, Bismillahirrahmanirrahim. Assalamualaikum and good morning, everyone. So thanks for the introduction. Again, let me introduce myself. I'm Dr. Awis Sukarni bin Muhammad Sabri. You can call me Dr. Awis. I'm from Department of Pharmaceutical Chemistry, the same department with Prof. Alfi, under the Kuliah of Pharmacy, International Islamic University Malaysia. So for today, I'm going to present regarding the 3D printing and emerging technology in pharmaceutics. My slide actually, because based on the conversation with Dr. Aina, I understand that the main audience is the undergrad student. So I make it's more like a lecture with some of the interactive activity. So it's going to be light and easy. And let's say if you have anything that you want to ask me, and feel free to stop me anytime. I'm fine with that. Oh, okay, let's start with the introduction. What is the 3D printing? So 3D printing or three-dimensional printing is a manufacturing process in method in which objects are made by fusing or depositing materials such as a plastic, metal, ceramic, powders, liquid, or even living cell in layers to produce a 3D object. So as you can see in this picture here, you can see here there's a jagged surface here. So this one is actually each of the layer of the printed material. So basically what they did is they start with the first layer. Once they done the first layer, they will print the second layer on top of the third layer, so on top of the first layer, and then after that they will continue with the third layer, fourth layer, and the fourth on until it's become the 3D object like this. So it was the earliest 3D printing actually was developed by Hideo Kodama in 1981. So the first prototype is basically quite simple. It's used the laser technique. So it's one of the 3D printer that is commonly used as well, which is I'm going to cover later on. So the theory is basically they use the resin, which is made from the photopolymer material, which is they're going to react with the presence of the light. So what they do, they will point the laser on the surface of the resin, and then by making that one, the photopolymerization process will occur and harden the resin. Okay, so there are a few types of 3D printing. So the first one is the binder jetting, and then there is direct energy deposition. There is a material extrusion, also known as the fused deposition modeling or FDM. So this one actually is this printer. And this one actually is the most common printer that you can find in the lab or in the office. And this one is the most commercially available as well. And it's quite cheap now. In Malaysia, the price is roughly 1,000 ringgit Malaysia. And the next one is the material jetting. And then there's the powder bed fusion, sheet lamination. And the last one is the VAT polymerization, or also known as the stereolithography or SLA, or also known as the laser 3D printer. So it's this one. But for this talk, I just going to cover three type of 3D printer only, because these are the most common printer that normally people use in terms of research. So the first one is the binder jetting, and then the second one is the FDM, and the last one actually is the laser 3D printer. So there are a few pros and cons of using this 3D printer. So let's start with the pro one. The first one is customizable. What they mean by this one is actually you can create any shape that you want. If you want to make it the tablet shape is like the standard the standard cylindrical shape, then you can make that one. Or maybe you can make it into a bear, for example, like a bear. Because this one is to encourage the children to take the medicine. So you can make it into something in that it's sort of like you kind of cheat the children to think it into it's a candy. So it's easier for them to take the tablet. The second one is the rapid prototyping. What they mean by this one is because, let's say if you want the conventional tablet, for example, if you want to make the tablet, if you want to make a certain shape, it means you need to prepare the mold first. So in order for us to make a mold, it's going to be quite expensive, and it's a lengthy process because you need to make the shape of the mold. So normally the mold is made from the metal, and then after that you have to basically create the mold based on the shape that you want. So but for the 3D printing, let's say you want to make it into a bear shape again, you just create the 3D image and then you can print it right away. So it's easier. You can change it. Let's say you don't happy with the shape, then you can straight away change the design, and then you can print it again. So it's a rapid prototyping. That's what we mean by this one. And the third one is print on demand. So the 3D printing is basically almost like the normal printer that you use to print something on the paper. So it means if you want to print something, then you can turn on the printer, click the file that you want and then print it right away. So you don't have to, so this one is mean that let's say certain material is a bit sensitive, so you cannot prepare it in the bulk. So what it mean, you can print the amount that you require only, so you don't have to print anything, and you can print any time that you want. And the next one is the greener technology. What they mean by this one is because normally for the conventional methods, you need to use a lot of steps. I mean, let's say for the granulation, you need to make a wet granulation. You need to put there is a binder, there is a filler, etc. And then there's a liquid that you need to add this one, and then you need to dry it again. So the process is very long, and then there is a lot of material that you add this one. But for the 3D printing, now mostly what you need is just the polymer as a carrier, and then the active ingredient that you want. That's all. So less material is mean the technology is going to be greener. And it's cost effective. Again, it's almost similar with the green technology. Like I said, the material that you're going to use is less, a lot less compared to the conventional. So once you cut the material, you cut the cost as well. And it's simple process. Like I said, in theory, basically you just mix the polymer with the API, and then you can just print it straight away. So because like a normal manufacturing process, like I said before, you need to use, let's say for the tablet, you need to do the granulation process, which is quite a lengthy process. So for this one, it's basically just to mix the polymer with the active ingredient, and then you are good to go. You can print it right away. Okay, next is the cons. So the first one is a limited material. What they mean by this one is actually because in order for you to print a certain material, the material need to have a certain properties. For example, they need to have the glass transition temperature. They can, because let's say if they only have the melting point, it's going to be running down like a water. For example, like a ice cube. Ice cube, they have the melting point. So if you melt the ice, it's become like a water. So it's going to running down. So you cannot harden it after that. So you cannot shape it. But if your material have glass transition temperature, this means it's going to become like a molten lava. So the molten lava is sort of like a solid liquid. So it is running down, but it's in a solid state. So you can shape it as whatever the shape that you want, and then you can cool it quite quickly, and then it's going to maintain the shape. So in order for us to print something, then we need this kind of material. In the next one is the build volume. What they mean with this one is, let's say you want to build something big. Because the printer is quite small, so the space is quite limited. So if you want to print something big, then it's going to be a bit tricky. So it means you need to build a bigger printer. So that's what we mean by the build volume. The next one is a slow process. To print some object using the 3D printer is going to take quite a long time. For example, like if you want to print this kind of shape, the phone, for this shape, normally I think it's going to take you roughly 2 hours, even though like this wasn't a big object, but it's going to be quite slow process. It's going to take you around 2 hours just to print one phone shape material, phone shape object that you want. Okay, now let's move to how to print the 3D printing. So because before we can print something, this is the most important step. You need to create the 3D object or 3D image that you want to print. So for this one, normally you need to use the computer aided design software to create this image. So the most common one is the Tinkercad. There is a SketchUp Make, FreeCAD, and many more. In my case, normally I will use the Tinkercad. Actually, it's a free software. You can use your own browser to use this software. Later on, I'm going to show you how to use this software to create one simple 3D image. And then you can use the MRI image and the 3D scanner as well, because when you print the MRI, they're going to print it layer by layer in a way. So it's similar with the one that we're going to create using the software. And after that, you have to save the file in the STL image. And then after that, you have to slice it. What I mean by slice is, let's say, again, if you want to print this phone, you cannot print it as one whole object like this. So what you need to be, like I said, you're going to print it layer by layer. So in order for you to print it layer by layer, so for this phone, you need to slice it into a lot of layer. So that's what we use this the slicer software. You need going to slice it, so you know that which one is the first layer that you need to print first, and then after that there's the second layer, third layer, and then so on. So let me show you how to print the how to create the image using the software. So this one is the Tinkercad. So you can just Google it, and then you just click this one, and then you can create the image that you want. So you just click use the 3D image. Okay, so the first one, let's say I'm going to show you how to print. Let's say you want to print a normal tablets. So you can just put this one. Normally the size, the normal tablet is a what I call it, cylinder shape. So, and this one is the measurement is actually in millimeter. So let's say, let's make it bigger. Let's say, let's make it, keep it at 20. So this means this one is 2 cm. And this one, just make it 10 cm. Okay, so easier for you to see. Okay, so this one actually is, that's how you're going to create the image. So you can choose all these pre-made shape here, and then you just click in and then drag it here, and then you can change the shape. And then let's say you want it to make it like a hole here. So you want it to make it like a donut shape. So you can click this one, the cylinder here, or just a normal one, and then change the size, depend on the size of the hole that you want to create. And then, so once you have this one, drag it and then put it in the middle. And then you choose here, hole. So it's going to change into a hollow object. And then you just click, drag it all, and then you can combine it together. So once you combine together, and then if you look at from here, you can see now it's become like a donut shape. So you can print this one. So let's say once you're happy with this shape, then you can export it, and then you can save it as the STL. Okay, so after you're done with this one, so you can go, you can download the Ultimate Cura, the software that normally use, and then you can just choose the object that you want, and go to the Ultimate Cura, and you just drop it. So from here, you can see this is the shape that you want, that you just prepared just now. So from here, this software is basically, like I said, is to slice the object. So so you can print it layer by layer. You just click slice here, and then from here they're going to estimate that, how long it takes for it to print. Even though this one, this quote unquote tablet here is just a 2 centimeter diameter, it's going to take us 40 minutes to print it. And then if you look at the preview here, then it's going to show you how many layers. For here actually there are 50 layers of this one in order for it to make the shape that you prepared just now. So if you go drag down, then you can see this one is the first layer. So they're going to print it, and they're going to stack it on top of that until it become a proper image, the full object. So that is the standard method that we use in order for us to create the 3D image. So again, this the summary of the process that I mentioned just now. So the first one, you're going to make the object, the shape. So once you make it and then you slice it, then you can then transfer it to the 3D printer, and then after that, you're going to print it, and then you get the image that the object, the 3D object that you just created just now. Okay. So moving on to the first printer that I'm going to cover. The first one is the binder jetting. So it's a 3D printing process where a liquid bonding agent selectively binds region of a powder bed. So it doesn't use heat. So for this one is basically, let's say you have the powder and then you already mix it with the active ingredient that you want. So what you're going to do, the first one, you're going to spread a thin layer of a powder. And then after that, you're going to drop a liquid bonding, which means it's a binder. So the binder acts as like a glue. So you're going to drop the binder on top of this powder. So, and then after that, you're going to apply another thin layer of the powder on top of the first layer. So at that point, because you already put the glue, so the glue is going to bond the first and the second layer together. And then you just repeat the process. You just drop the binder on top of the on top of the powder, and then stack another powder layer on top of that. Then you're going to repeat the process until it becomes a 3D object. So it's more a printer over the powder surface depositing binder droplets. So this one is similar like you print something using the normal printer that you have at home, the one that you print on top of the paper. So literally it's going to print like that. But instead of printing an ink, they're just going to drop the binder on top of that. And then since we use the powder, so the powder need to be a very fine powder. So typically it need to be roughly around 80 micrometer in diameter. So because if the powder is quite big or quite what this mean, it's like a granule. So the shape that you're going to get is not going to be smooth, because if the powder is a granule, so it means it's big diameter. So the shape on the surface is going to be bumpy because of the size of the powder. And then in terms of the binder, normally you will use the normal binder that you use when making tablets. So normally the common one is polyvinyl pyrrolidone, PVP, and then you can use the HPC and PEG as well. HPC is hydroxyl propyl cellulose, and PEG is polyethylene glycol. You can use that one. So normally they use water soluble binder because it's easier for you to dissolve it. And so, and then like I said, the droplet will bind this powder together, and then it's going to create the 3D image that you just created just now. So again, this one is the example of the diagram. So the first one is going to spread a single layer, and after that it's going to print. So this one in this case is going to print the tablet. So it's going to create the shape, and then after that it's going to spread another layer of it on top of this one. So let's see if I can open this.
Two-dimensional printing, or 3DP, was originally invented in
So this one actually is one of the demonstration video from Labra University. So they printed a shape of a car, a sport car, but the concept is similar with the
MIT
binder.
in 1993. A year later, there's a maximum build volume of 203 by 254 by 203 millimeters. The 3DP process begins with the creation of a 3D CAD model. This can be done in numerous programs, including NX, SolidWorks, and many more. Upon completion, the model is converted to an STL file that is able to describe the surface geometry of the part by translating it into a number of small triangles. The STL file is then sent to the ZPrint software. The 3DP process allows full-color models to be produced, providing an aesthetically pleasing finish. Build software slices the part into layers 0.089 millimeters in thickness and sends it to the printer. The roller spreads a layer of ZP130 plaster powder over the build pan. The printhead then moves across and prints a layer of binder solution infused with colored ink in the shape of the first layer. This binder solution glues the powder together in the desired shape. Any excess powder left unbound acts as a support structure for the part, much like the SLS process. The platform moves down a single layer thickness, and the roller then spreads another layer of powder across, thereby gluing it to the binder below. The printhead once again moves across and expels the binding agent in the shape of the next layer. This process is repeated layer by layer until the model is complete. The part is removed and placed in the post-processing chamber, where a high-powered jet of
Okay, so basically, because I just choose this video because it's easier for you to imagine, for you to see how this process rather than imagine how the process is. Okay, so the one of the product that they use the binder jetting in pharmaceutical application is actually this tablet is a Spritam. It's actually the first and the only FDA approved 3D printed tablet in the market. So this one, the active ingredient is levetiracetam. It used to treat the epilepsy. So by making this tablet into using the binder jetting, they managed to improve the discretion properties of this tablet, because this tablet is basically like a normal tablet. Normally you need to compress the tablets. So when you compress the tablet, they will affect the disintegration process of the tablet. But this tablet is basically, it's just a powder that been glued together. So they didn't compress it, they didn't melt it together. So it's just a powder that been stuck together. So when you do that, so if you look at if you use the SEM and then you look at the surface of the powder, you can see this powder actually is very porous. So when it's porous, it means you increase the surface area of this tablet. So by doing that, you will improve the solubility of this powder. And on top of that, because the binder that they use is water soluble. So when you drop this tablet in the water, or maybe when you take it in the stomach, so it will rapidly disintegrate because it's just a water soluble. So it can easily improve the discretion of the tablet. And then after that, of course, once you improve the discretion, you will improve the dissolution time of the tablet as well. And then move on to the next printer is the FDM, or the fused deposition modeling. Again, like I said, the FDM are the most commonly available and affordable type of 3D printer technology globally. I think this one you can get the cheapest one as well. The one that I use in my current lab is Ender. You can get it. I'm not sure the currency conversion for Malaysia ringgit to the Indonesia rupiah, but for in Malaysia you can get it roughly around 1,000 ringgit Malaysia. And then even the ink or the filament that you're going to use to print, it wasn't that expensive. It's one roll is around 100 ringgit Malaysia. So, and then one roll can last a few months. So this technique is basically used, like I said, the filament. The filament is something, is look like a spaghetti shape. It's literally like a very long spaghetti-like ink, quote unquote ink. And then so basically you're going to use this filament, you're going to attach it to the printer, and then it's going to fed through the printer nozzle in the extrusion head. So in the extrusion head, or the printing head, or the printing head is basically where it's going to apply the heat, and it's going to melt the filament that we have. So by melting the filament, it's going to, and after that it's going to move around the surface of the platform. So it's going to drop a bit by bit. So it's going to start to print layer by layer. So this process is basically the one that you can see on my second slide just now, where they're going to print layer by layer until it become like a 3D printing shape. So in the printer, actually in the nozzle, like I said, is where they're going to heat the filament. And then after that, there is also a motor to push the filament through the heated nozzle. So it's basically, it's going to push the filament through this nozzle while they move around while they're printing the printer. So by keep on repeating this process, then you can create the 3D image. So the concept is almost similar with the binder jetting just now. It just, instead of using the powder, this one they use the filament itself as an ink. Okay, so this one is the example of the FDM 3D printer. So this one is the one of the example, and then this one is the roughly the diagram of the printer. So the concept of all the printer is going to be the same. They have the feeder. This one is basically where they're going the motor that they put to push the filament through the printer head, or this one, the hot end here. So, and then this one is the printer head where they're going to melt the filament, and then this one is the printing bed, so where they're going to print the model layer by layer. So the printer, the process is basically this printer bed is going to move back and forth, and then the motor and the printer head here is going to move side by side. And then of course they have this one, the positioner, is where it's going to move up and down. So normally in the process, the position is going to move layer by layer. I mean that the height is going to be one height layer by layer. And so in the pharmaceutics, because this printing basically is going to be the same. So what the most important one actually is the filament, because in the market, the one that they sell is there are PLA, polylactic acid, there even a metal, there even a wood type of filament. But for us in the pharmaceutics, we need to create our own filament. And so in order for us to create this filament, so you need to use one technique, we call it HME, or hot melt extrusion technique. Okay, so what is the hot melt extrusion? It's basically, it's quite old technique. Is was established in early 1930s, and normally it was used in the plastic production or rubber at the food industry as well. And then, but in beginning of 1970s, the researcher in the pharmaceutical industry start to apply this technique in order to formulate and produce a new, it's not a new, it's something, a control release powder, because when the technique is basically is a mix and melt technique. So you're going to mix the API or the active ingredient with the polymer, and then after that it's going to melt it together, and then it's going to combine it together. So by doing this, you can first you can control release the formulation that you want. So it means the tablet that you're going to produce, you can control it, either make it fast release or control release or entry release as well. Then after that, when the 3D printing become like a quote unquote a new technology in the pharmaceutical field, then they start to apply this technique in collaboration with the 3D printer. Because in this, because you need to create your own ink. So in order for you, if you want to use the FDM, in order for you to create your own ink, then you need to use this method, this technique. So there are this technique I'm going to show you later on. So basically, there are screw that you're going to attach in the machine. So there are 3 types of screw. There is single, twin, and the multi screw extruder. So this one is the cross section of this single and twin screw. You can see here, this one is let's say if you cut it the machine in half, then you can see this one is the hopper. It's basically this where you're going to insert your sample, and then this one is a screw. So if the single screw literally is just a single, and then the twin screw is going to be 2 screws. And then this one is the orifice or the nozzle where you attach the nozzle. So this one where you're going to control how thick that your filament is going to be. So normally for the 3D printer, the size is 1.75 millimeter. So let's say you put in your sample, it means the polymer and your active ingredient together. And then when you put in here, so it's going to mix it together. And then because at this chamber, they're going to heat up the sample. So basically at this point, it's going to start to melt it together. So they melt all the normally they melt the polymer, and then the API will be suspended into the polymer. So they mix it together, and then they pump it out through this orifice, and then you will get the filament. And for the twin screw, the screw, they can move to one direction or opposite direction. It means if they move to the one direction only, so it means let's say the screw is going to move the anti-clockwise, so both of them are going to move anti-clockwise. So this one is basically if you want to speed up the process. But let's say you want to improve the mixing process, then the screw is going to move the opposite direction. So it means one of them is going to move clockwise and the other one is going to move anti-clockwise. So by making that one, you can mix the mixture better, because basically you're going to push it together, you're going to press it together while melting it, and then after that you going to eject it and it become a filament. Okay, so this one is one of the example here. Let's say this one, your API and your filament, you put it together, and then at this one, this screw is going to act like a conveyor. So it's going to push it through here, and then it's going to melt and mix it together. And then after that, it's going to push, and then you get this one. This is what we call it the extruded, or we call it sometimes we call it filament. So this filament is the one that we're going to use to attach to our printer. Okay, so but this technique is as in the name of the technique is hot melt extrusion. So it means it's hot and melt. So it's going to apply the heat in the sample. So let's say some of the active ingredients are very sensitive. For example, even the extract, let's say you have the plant extract. Some of the plant extract are quite sensitive to the heat. So in order for you, let's say you have the plant extract, and then is the degradation point of the sample, let's say it's 100 degree. So you need to use the polymer that have a low glass transition temperature. So for example, like polyethylene glycol, the transition temperature are roughly between 40 to 50 degree. So you can use that. Yeah. So and then you combine with your API or your active ingredient or your extract, and then you can extrude it without degrading your active ingredient. So in order for you to know what is the degradation point of the active ingredient that you have, then the first one you can use the DAC or the TGA. Differential scanning calorimetry, or you can use the thermogravimetric analysis. So by using these two instruments, then you can determine what is the degradation point. So degradation point is basically where you burn the sample. So there is no more turning back at that point. So once you exceed the degradation point, then your sample is already ruined, so you cannot use the sample. So so you just take a small amount of the active ingredient that you're going to use, then check it what is the degradation point of the sample before you can use is on the hot melt extrusion. I do attach here the link of this machine, but it's basically just like this one. So if I can share this slide, and then you can have a look at the link that I attach here. So these are a few of the characteristics of the material that can be used for the in the HME for the pharmaceutical application. So the first one, they must meet the same level of purity and safety as those used in the traditional dosage form. This means it need to be pharmaceutical grade, because they say that if you want to print the tablets, then the powder or the polymer that you use need to be pharmaceutical grade. And then the next one, you must be able to deform easily inside the extruder and solidify on exiting it. So in this one, like I said before, the polymer or the carrier that you're going to use need to have a glass transition temperature. So it means it's going to soften. The right word is actually soften. It's not melt. It will soften the material, and then upon the exit, they can blow the cool gas. The cool air. So upon the exit, it start become hardened, so you can retain the shape. And then the other one is you need to look at the what do you call it? The viscosity of the sample. Let's say if your sample you have a very high viscosity, then it's going to be hard for you to extrude it, because when normally the sample with high molecular weight, because since they have a very long chain of the compound, then it's become too viscous. So once it's become too viscous, it's going to block the HME. So and then so you cannot extrude the polymer. So it's gonna come out from the extruder. And then the third one, there must be thermostable and maintain an acceptable physical and chemical stability during the HME process and afterward during the long-term storage. This is what I mentioned just now. The sample that you need to use need to be able to be stable at a certain temperature, because again it's going to apply heat, and then so if your sample are very heat sensitive or maybe the degradation point are too low, then you cannot use the HME technique for the sample. And the next one, the last one, is the desired in vitro release and in vitro performance should be achieved by the final dosage form. This means, let's say by combining this one, let's say sometimes you can control release, like I said before, you can control release the dosage form. Let's say once you mix it together, and then the final dosage form, it should be showing what your target is. Let's say your target is going to be a slow release. This means after you extrude the filament, and then you print it into a 3D printer, then you test it, it should show this kind of properties at the end. Because sometimes when you physically mix this material, they might show these properties, but when you apply, when you put it in the hot melt extrusion, it won't show this kind of properties. So that's why for this kind of technique, you need to check it before and after of the process. So it means before, which is the physical mixing, and after you extrude it, does it show the similar properties that you aiming for? Okay, so this one is basically when you combine these two techniques together, the hot melt extrusion and the printer. So again, this one is the API, or your in your case you can put your plant extract as well, and then this one is the polymer where it's going to act as a carrier. And then so after you put it in the hopper, and then you're going to mix and melt it, and then the filament that you get is become like this. It means the active ingredient or the API here is going to be suspended on the polymer, on the molten polymer. So the polymer is just going to act as a carrier. And then after that, you're going to create the 3D image like I showed before. And then after that, you're just going to attach this filament to the 3D printer. So when you attach this one, so at this point here at the 3D printer head here, it's going to print your sample using the filament or the ink that you just prepared just now. And then from here, then you will get the 3D printed dosage form that you prefer. Is either it's a patch or maybe it's a tablet that you want. So these are some of the sample or the study that been done using the FDM in the pharmaceutical application. So this one actually is the current study that been done by my student with my colleague. So this one is basically my student tried to try to print the isoniazide tablet. So in here you can see this filament here. There are isoniazide plus the hydroxypropyl cellulose and the polyethylene glycol in the sample. And then after they got the filament and then they print it. Here actually is the first tablet that she managed to print. So you can see it's quite rough here. And then now actually there is a new one that she just printed, and at that point she got a lot better shape of 3D printed. But here you can see it's quite rough. But the one that she just printed last week actually, the one that is have a very good shape of a tablet. So in this case, the HPC here actually is the carrier, and the PEG here is act as a plasticizer. So because HPC is a very high viscous, so when my student at first she just printed using she just extrude the sample using the isoniazide and the HPC only, and at that point she found that some of the sample are quite hard. quite difficult for you to extrude, and then after the extrude, the sample become is too elastic. So it's just like a rubber. It become like a rubber. So the thing is, when it's become like a rubber, because if you remember, like I said, there is a motor to push the filament from the 3D printer. So because it's too soft, so they cannot push it. When they push it, it's just going to, it's sort of like you push cloths. So it's just going to fold at the entrance of the printer. So you cannot really properly push the filament. So that's what, at that point, I suggest her to include the polyethylene glycol in the formulation. So by this one, it's going to improve the texture of the filament. So at this point, you can see here, the filament is still very flexible, but it's not that soft. It become almost like a plastic kind of feel. So it's become hardened. So that's why after she managed to extrude this one, then she managed to print the tablet. So this one is the ongoing study. So the result is still ongoing, and I cannot show all the result, but I can show you the result from the other study that been done by other researcher. So the first one is the study done by the Campaign et al. The title of the the journal is Immediate Release 3D Printed Tablet Produced via Fused Deposition Modeling of a Thermosensitive Drug. So in this case, what they did is they studied the printability of the thermosensitive drug. It means the drug that have a low degradation point. So as you can see here, the filament that they use, this is the polymer that you use for the extrusion, and then they use the polymer that have a very low transition temperature here. You can see here, roughly between 40 to up to 60-ish, and this one is just a control. So they decide to use one with the higher melting point here. So this one is the highest one, 142 to 145, and the rest are below 60 degrees Celsius. So it means, let's say the degradation point of the API here, let's say if the degradation point is low, then you can use this material. And then after that, they managed to extrude most of the filament here. You can see the filament here is quite nice, and then from here you can see this one is basically the API that is be suspended on top on the filament. And from here, the Collicot IR, because like I said, this one is the only sample here that have a quite high temperature for the melting point. So you can see the color are quite dark. So which means the sample or the active ingredient are already degraded at this point. So that's why it showed like a charred color, a black color here. And then so from here they choose the best one, and then they printed the tablets, and you can see here they showed a quite nice shape, except for this one, the polyxymer. The shape is a bit wonky, but for the best one is actually the PVP K12 here. It's show a very sharp image here, followed by the PEG 6000. And then from the study, they run it using the SRD to see if the drug changed from crystalline to amorphous, because the drug that they use in the crystalline shape, is in the crystalline properties. And then you can see here, if there are crystalline, they will show you a peak here like this. But when at this point here, when you mix it with the PEG 6000, and then this one is the physical mixture, and then this one is where they mix it using the HME. From the physical mixture, you still can see there is the crystalline properties of the drug. And from here, you can see some of the peak are gone. So it's mean, but there's some of the peak is still retained. So it's mean at this point, it's already become polycrystalline or polyamorphous. And for the PVP here, you can see the peak are totally gone. So it means the crystal now, the drug now it become amorphous. The thing is with the drug, when they are crystalline state, they are stable, but they have a poor solubility. But when they are in the amorphous state, it means they are less stable, but they are very have a very good dissolution properties. And here you can see the release profile of all of the tablet that they printed. And then so this one is the powder, and then this one is the mixture of the PVP. And you can see here when because this one the drug already changed from crystalline to amorphous state, so the release profile are very fast here compared to the other one, because for like this one is the drug is in the is a semi-crystalline or semi-amorphous, but here when it's changed to the amorphous state, you can see the release profile are way faster compared to the other drug. In fact, in this study, they mentioned that this tablet are so far the fastest release profile among the 3D printed study that been done by other researchers as well. And then this one is another study done by Chaudhary et al. 2021. The title of the research is extruded effluent derived 3D printed medicated skin patch to mitigate destructive pulmonary tuberculosis designed to delivery. So this one is basically what they aim to make a patch here, and then where they loaded quercetin with the PVP in the patch. So this one, the first one actually is the one with the quercetin, and the second one is just the blank. It means it's just the polymer without the active ingredient. And you can see here when when you add the active ingredient, sometimes they change the color depend on the color of the active ingredient. Because this one, I believe the color of the active ingredient is slightly yellowish. So you can see here this one is more yellowish compared to this one, because normally for the polymer they tend to be a milky color. And then from here you can see the in vitro study. The patch showed like a constant release up to 70 hours. You can see the release is quite consistent, except for the T1, the T1 formulation, where they showed a quite high release profile at the beginning, and after that it start to release at a constant rate. And then so this one actually they tested the patch on the rat. So when they put the patch on the rat, at first around almost 200 hours here, before the 200 hours, they couldn't find any, they couldn't detect any active ingredient or the quercetin in the rat plasma. But after the 200 hours, then they start the increment of the quercetin in the rat plasma. So it means the patch managed to penetrate the skin and then enter the body system of the rat. And then up to here, and you can see it's maxed out around 300 hours here. Okay, so that is the study that used the FDM. And then the last one that I'm going to cover is the printer that they use, the stereolithography, SLA, or also known as the laser. So SLA printer, basically they use the mirror known as the galvanometer or galvos, with one position on the x-axis and another one on the y-axis. So what I mean is, below here there is a mirror here and there is a mirror here as well. So what they did is basically they're going to point the laser at this mirror, and then the mirror is going to reflect to the surface of this sample here. As you can see here, from here, this one is where's the laser aiming at the surface of the resin. The resin is here is basically a photopolymer resin. So when this light been pointed or been lasered to the when the laser will been pointed to the surface of this resin here, then photopolymerization will occur. So what I mean is the polymer start to become hardened, and then so it's going to stuck on, or they're going to start on the surface of this material here. So as you can see here, after they print it, and then they're going to lift up, and then they're going to scrape the surface of this container. This one is because if let's say they just keep on print the laser here, the first one, the object here is going to stuck at the bottom of this container, and then the other one, because you want to put a layer of a new uncured resin. So in order for you to print the next layer. So that's why after they print print this one, the first layer, one layer here, and then they're going to lift it up, and then they scrap it, and then when they lower it down, they're going to lower it slightly higher. It's a one layer thickness compared to the previous one. So that's how they're going to print it layer by layer. Like I said before, the SLA uses the photopolymerization process. So basically, they use the visible or UV light, but the most commonly is UV light to initiate and propagate polymerization reaction to form a linear or cross-linked polymer structure. So basically, in the resin, there is a monomer, oligomer, and the photoinitiator as well. So the photoinitiator is basically act as to absorb the energy in order for it to induce the polymerization, the photopolymerization here. So here, once the photoinitiator absorb it, and then they start to create this, they're going to link, they're going to start to create the bond between the monomer and the oligomer here. So this one is the example as a monomeric styrene and the oligomeric acrylate. When they put it together, and then after that they add the photoinitiator. So once the photoinitiator, they absorb the energy, and then they start to create the new bond here, and then now it's become one polymer, one big polymer here after being photopolymerized through the process. So before we can print the sample, because you can buy the resin in the market, the ready-made one, but that one there is no the ingredient that we want, especially the extract, or maybe the extract, or maybe the active ingredient or the drug that we want to add in the resin. So so this one, what you can do is basically you just need to prepare your own polymer first. It means you need to add the photopolymer, and then you need to add the photoinitiator, and then you need to add the other ingredient that you prefer. For example, for the photopolymer, the common one is PECDA, is polyethylene glycol diacrylate. That one is one of the common photopolymer. And then you can add the photoinitiator. There are a few different type of photoinitiator, and then you can add the active ingredient that you want, and then you can take a bit of it, and then you need to test it first, because you cannot simply start using it on the 3D printer, the laser 3D printer or the SLA 3D printer, because let's say your mixture doesn't really react with the light, with the laser, then you're just going to waste the sample, because it's not going to print. So what you can do is you can take a bit of the sample and then put in the container or something, and then put it under the UV light, and then you can see if the sample start become hardened. And if let's say if you have a good microscope with the digital camera, then you can put it on the microscope with the digital camera, and then you can record the process, and then you can put the UV light next to the microscope and then record it. So normally when the photopolymerization occurred, is sort of like if I think you're familiar when the water become ice, so they start to show like a mark where they start to solidify. It's sort of like a how to say this one? It's sort of like when you watch the Christmas movie, then it's like when you see there's a water and then it's become like an ice shape. So similar, almost similar with this one. When the photopolymerization occurred, then you can see it become hardened and they showed you a pattern of a solid sample from solidified sample from the liquid. And so you need to see how long it takes for it to photopolymerize. If it takes longer, then it's not suitable for the 3D printer. It should be occurred within a few seconds. If it's photopolymerization occurred within a few seconds, then you can use the resin that you prepared and then using the 3D printer, the laser 3D printer. So this one is some of the example. Actually, this one is from my PhD thesis when I did my PhD in 2012 until 2016. So this one is actually a 3D printed chloramphenicol ocular mini tablet and insert. The idea of this study is basically because when you apply, let's say you have a problem with the eye, and then the normal treatment is basically you're going to apply the eye drop. The thing is, when you apply the eye drop and then you automatically start to blink. When you blink, so basically you remove most of the drug from the eye. So in other words, you reduce the therapeutic level of the drug. So and then again, you have to apply it a couple of times a day. So when you have this the ocular mini tablet or the ocular insert, so basically for the mini tablet, the idea is for you to put it on the front of the eye or the end of the eye, and then leave it there. And then once they interact with your tear, they start to dissolve and then start become like a gel. So you just leave it there and then you start to slowly release the drug. So the idea is for you to you need to apply it once a day. So you put it there and leave it and then start to dissolve slowly. So you don't have to keep on reapplying the drug. And for the insert, the target is actually for a very long duration. Let's say you need to apply a drug and then you need to apply it for a few days, for example. And then the idea is for you to put the insert actually under the eyelid, so you can lift up the eyelid and then basically you need to in a way shove in the insert under the eyelid and then leave it there. And then basically the idea is start to dissolve slowly. So it's going to provide with the drug level that you need for a very long duration. So as you can see here, this one actually was made using the resin that I prepared, and this one actually is the mini tablet that been prepared using the normal punch. So it's a 2mm tablet punch. As you can see here, but for the normal tablet, you can see the shape is quite nice. It's like a normal tablet. I have a very nice sharp shape. But for the printing here, you can see on the top is fine. At the side is fine as well. But at the bottom there is what we call it a skirt. This means you can see here there is a bit wider compared to the top. So this one is quite common in the laser printing, especially if you prepare it using your own resin, because at this point, this one is basically at the surface of the platform that you prepared. So compared to the resin that you buy from the market, normally it's going to show a very sharp one, but this one, because there is a chloramphenicol, which this drug basically slightly interfere with the photopolymerization process. So that's why you get this kind of skirt here. And at the beginning of my study, I did mention that you can print using the MRI image. Actually, during my study, one of the lecturer, for some reason, he got his head scanned using the MRI scanner. He said it's for fun because actually he want to use the MRI image, and then he literally printed his own head using this MRI image. But in my case, I don't need the whole head, so I just need the size of the eye socket. So I just I asked his permission to use his MRI image, and then he just gave me his MRI image. And then so from here, I look at the axial, sagittal, and coronal plane view, and then I measured the eye socket here. So, and then from here, then I can create this ocular insert here based on the shape of the eye. So it means this ocular insert is going to be customized based on the patient itself. So if because some of the patient, they have slightly bigger eye socket or slightly smaller eye pocket compared to the other one. So this one is basically a customized ocular insert. And then after that, I just create this insert here based on this MRI. And then I tested on the drug release. And then as you can see here, for the the first formulation using the polyethylene glycol 6000, TEG 6000, and then so this one is the polyethylene glycol only, and then you can see here the release profile are quite faster compared to the other one. So this one is PEG 4000, and then this one is just the drop without the PEG. So you can see here with the addition of the PEG 6000, it shows a faster release profile, and then after that it start to plateau. It means at this point, they already released most of the drug. But here you can see the release profile wasn't as smooth as the PEG 6000 here. For the PEG 6000, you can see it's quite nice curve here until around 24 hours. And then it start already release most of the drug, and then it start to plateau. But here you can see it's released slowly. It's released slowly, and then suddenly it jump here quite fast here. So this one is because at this point it start to dissolve slowly, and but at between the 10 hour and 24 hour, they start to crumble, and then so they break into a smaller pieces. That's why at this point, the release profile are quite fast, because at that point the tablet, they just crumble, they just break into a smaller pieces. Compared to this one, they just dissolve slowly without breaking the tablet. And then so this one is I just combine between the mini tablet and the insert that I printed. So you can see here for the mini tablet here, they showed a faster release profile compared to the insert, except for the PEG 6000 here. You can see here the release profile basically is similar, slightly faster at the beginning, but after that it becomes similar. It's just the same release profile compared to the other one, whereas the mini tablet showed a higher release profile compared to the insert. Again, so this one is because for the insert here, for the PEG 6000, it showed a very smooth release profile, but for the mini tablet here, because they break into a smaller piece, smaller piece, so that's why at that point it released faster compared to the insert. Okay, so this one actually a study done by other researcher from the Martinez et al. 2017. So in this study, the study is fabrication of a drug-loaded hydrogel with the stereolithographic 3D printing, or the SLA. So in this study, so basically they printed the hydrogel, where they incorporate the riboflavin and thietanolamine as the photoinitiator. And as you can see here, this one is without the water, and then this one is the 10, 20, and 30%. So basically the shape is almost the same. But when you run, when you look at the DSC, and you can see here, with the 0 and 20%, the melting point of the polymer doesn't really show here, but for the at the 30% of water here, you can see there are a very big peak here. So this one actually is because of the water. That's the thing. If you want to run the sample using the DAC, you need to make sure your sample at least there a very small amount of water. It's better for you if there is no water at all, because if there is water, this peak actually is the water that been evaporated from the sample. So let's say if you have any peak in between, since you have the big water sample here, so it's just covered the transition temperature of the sample. So let's say if you have like this one is a very high percentage of water, what you can do is basically you need to dry your sample first, or in this case you can heat up the sample up to 100 or 120 degree, leave it for a while, and then cool it down, and then rerun it again. So it means the first process is for you to remove the water, and after that is the actual process where you're going to see if there is any transition temperature happen in somewhere in this range. And then here you can see the release profile here for the 20 and 30% of water. The drug release are faster compared to the 1% and 0% of water. So this one is basically it means the presence of water does improve the solubility of the drug in the formulation. But the 20 and 30% of water, it shows a similar release profile. And for the for the 10%, it's slightly slower, and for the 0% is a lot more slower compared to the rest of the formulation. Okay. Now the last one is the evaluation of the 3D printed dosage form. So because this one, people might query if you print because the 3D printed is quote unquote sort of like a new dosage form. So sometimes they are not really sure what kind of the evaluation that we can do, the physical and chemical the evaluation that we can do for this 3D printed dosage form. So since the dosage form basically, even though it's a 3D printed, but the dosage form are basically the same with the one that in using the conventional manufacturing process. So for the evaluation, you need to follow the pharmacopoeia guideline as well. So the normal one is a BP or USP, or you can use the EUP. So if your dosage form are tablets, it means the 3D printed tablet need to go through the weight uniformity, the content uniformity, the distillation, distribution, the variability test, that kind of things. So and then similar with the patch as well. If it's use a patch, then the test need to refer to the pharmacopoeia guideline. And then the additional test, actually this one haven't been spelled out, but if you have this test, it's better because they can give you extra info regarding the study that you've done. And the first one is the scanning electron microscopy, or the SEM. This one because sometimes when you print the sample and you look at the sample, it look very smooth. But when you look closely on the surface, then you can see it wasn't that smooth. Maybe it's rough, or maybe it's a porous sample. So that's why it's good for you to have a SEM image of the sample, so you can really study the sample. Because sometimes when after you 3D printer it, you find it, the release profile, for example, is faster. And then after you scan it using SEM, then you notice, oh, actually it's because your sample are very porous. So that's why the release profile faster compared to the normal one. And then the next one is DAC, or differential scanning calorimetry. This one is because especially if you use the hot melt extrusion technique, sometimes when you mix it together, the drug, like I show you one of the result, one of the study done by the other researcher. Actually, at the beginning, maybe the drug have a melting point, but with the after you do run using the hot melt extrusion technique, the melting point disappear, and it become the glass transition temperature. So it means the drug or the active ingredient change the properties from the crystalline to amorphous. Or sometimes when you run it together, it means when you mix together with the polymer and then you run using the DAC, you can see the polymer have the transition temperature, for example, 150 degree, and then let's say the drug have the melting point at the 100 degree, for example. Once you combine it together, and then you find the melting point suddenly disappear and the transition temperature move slightly lower, maybe 140 instead of 150 degree. This one is because sometimes the drug itself will affect the transition temperature of the polymer. So that's why it's good for you to have the DAC study. And for the SRD, it's almost similar with the DAC. So basically, you want to see the drug, if the drug is from the crystalline is changed to amorphous or the other way around, or that kind of things. Or the other things with the DAC, I forgot to mention. Sometimes we want to see the degradation of the drug as well. So let's say sometime when you combine after the HME, you maybe accidentally degraded your drug because you're applying a too high temperature. So that's the other point of the DAC. And for the SRD, basically to see if the drug change properties from the crystalline to amorphous or the other way around, or maybe become the semi-crystalline or semi-amorphous. And for the FTIR, it's basically you want to see, it's almost similar with the SRD, the crystalline or the amorphous. And sometimes the other one, you want to see if there is any interaction between the drug and also with the carrier that you use. So this one, normally for this test, you need to do it with the physical mixture first, and then after that you need to test the sample that already gone through the hot melt extrusion technique. Okay, so by doing this test, then you can see if there is any interaction. If there is any interaction, then you need to find a new carrier or new polymer that doesn't have any interaction with the active ingredient, the extract or the drug that you use. Okay, so with that, thank you for listening. And yeah, actually, if you have any question or any, if you want to collaborate with me, you can contact me easily with my email, awisabri@ium.edu.my. And yeah, I'm open for the Q&A. Thank you.
Thank you. Thank you very much, Dr. Awis, for sharing the knowledge in 3D printing as well as your research. It was very exciting to learn what 3D printing technology has brought to us in the pharmaceutical field and with the virtually limitless application. Now we'll go ahead and take some time for questions. For all the audience, please raise your hand and then I will let you say your questions, or alternatively you can type your question in the chat box. Okay, there is no question yet for now. I would like to remind that you can also ask your question in Bahasa Indonesia and we'll try our best to translate your question. Or if you have fluent in Malay, then maybe you can ask your question in Malay as well. Okay, actually, I have a quick question, Dr. Awis, if you may. From what we have learned so far today, the filament feed or the polymers used for the 3D printing is majority from synthetic origin. For example, the HPMC, it's a modified cellulose, or the polyethylene glycol, or even the polylactic acid, glycolic acid, or PLGA, PGA. While they are biocompatible, with our body, is there a chance to use biopolymer, for instance, chitosan or alginate that are quite abundant in Indonesia, maybe in Malaysia as well, to make more biocompatible or even more biodegradable polymer products? And what's your take on it? And yeah, maybe some pros and cons, or maybe you have some experience in using them. That's my question. Thank you.
Okay, thank you. It's a good question. Actually, for the polymer, you can use any polymer. But again, it depends on what is your target. Let's say for the what you just mentioned just now, the polymer, I forgot the name. Alginate.
Which one?
Yeah, alginate.
Yeah, chitosan or alginate. Oh, sorry, my camera was off all the time.
The alginate, actually, you can use that one. And in fact, there are other studies that use the alginate for the 3D printing. Yeah, it's quite common. And then the other one is, you can, let's say your target is the biocompatible material, then you can use the alginate. And then the other one is the PLGA. You can use the PLGA as well. In fact, there is a study use the PLGA, and then after that they make the dosage form, and then they put it in the wound, actually, because so basically they put it inside the wound, and then they saturate the wound with the polymer inside the wound. So the idea is for the polymer to slowly degrade and then release the drug. The good with the for example, the alginate or PLGA, I'm not sure, but I'm quite confident with the PLGA. Our body doesn't reject the polymer. It means it doesn't register as a foreign substance. So if you put it inside your body, it won't show any rejection kind of symptom, rejection symptom. So it means it can easily what do you call it, it can easily degrade. Yeah, it can degrade kind of in the body without being rejected by our body.
Have you got any experience in doing with biopolymers?
I have one, but I didn't use the 3D printer for this one. The one of the study is basically I used the alginate to make the alginate bead. So that's actually one of the study that have been done by me and my colleague. We make a habatu sauda, it's the black seed oil. So we combine the black seed oil with the alginate, and then so we make it into a small bead. So the idea is for it to mask the taste, and then the other one is it's easier for the kids and for the older people to take the dosage form, because normally for the black seed oil they put it in the cap, and then so the capsule, and it's sometimes it's quite difficult for the kids or for the older people for them to consume it. So what we did is we make it into a very small bead. It just, I think the size is a few millimeter only. I think it's around 2 millimeters diameter. So the first one, you reduce the size. So it means by making it into a capsule, making it into a bead, and then you actually for the kids, you can just sprinkle the beads on their food, and then you can give it to them. So and then the other one is because they mask the taste of the black seed oil. So again, the kids won't taste the black seed oil. So so that one, but that one we didn't really use the 3D printer for that one.
Okay, thank you very much. Maybe from other participants. Okay, so can I follow up a little about this?
Yeah, sure.
It's now that I think about it, like we use 3D printing to make an interesting shape or a nice design of the formulation at the dosage forms, and then what kind of size that you envision the final product will be like? Will it be small tablet, or will it be a little bit big so that you can see the shape? And but in related to that, then we may think back about the potency of the drugs. Like you cannot just make a super big tablet using 3D printing. Otherwise, that would be inefficient because you need to use all the electricity and power and energy to make that. So does it have a certain limit for what type of drugs? Like it has to have a separate potency, or example, the high potency, or that we can just use the herbal products or natural products, because natural products we have to make it super concentrated to get a high potency. And so yeah, could you elaborate more in that regard, please? Thank you.
Okay, good question. In terms of the size, actually it depends on your target, the patient itself. Because there is a study done regarding the ability of the children to swallow the tablets. So they said that for the children, actually as small as 3 years old, they can swallow the tablets size 2 to 3 millimeter diameter. So it means for the kid as young as 3 years old, if you give a mini tablet, we call it mini tablet, then most of them will be able to swallow it. But if for the normal like an adult, I think 1 centimeter should be okay. It should be okay for them to take or to consume the tablets. In terms of the drug itself, like I said, the good thing about the 3D printing is basically, let's say for the other like the conventional tablet, for example, for the conventional tablets, then there is a lot of other excipient that you will need to add in the formulation. For example, the filler, the binder, etc. But for the 3D printer, basically you just need your extract and the what do you call it, the binder, so the polymer as a carrier. So there are a few studies that they managed to incorporate 30% of the drug in the formulation of the 3D printer. So it means for this one, if you want it to be a high amount of the active ingredient, then it's possible for you to go up to 30% of the whole of the whole formulation in one 3D printed tablets or dosage form. And the other one, actually, we have actually we just proposed, presented one of our research. Basically, we incorporated the extract from one of the plant, and then we're going to make it into a patch for the wound healing. So it's possible as well. And then actually there is a study done where they're making a patch, incorporated active ingredient or even the plant extract, because I believe for your school, your focus is more on the herbal medicine, that kind of target, right? So it's possible for you to incorporate the extract from the plant and then make it into a patch for the wound healing. That one is for sure. There are a couple of studies that managed to do the 3D printed patch with the herbal with the plant extract. So I think they're the one that you can maybe in future you can try to venture toward that kind of dosage form.
Thank you very much for the insight. Thank you.
Mungkin dari saya, Bu Irna.
Oh yeah, yeah.
Dari saya, Bu Irna, ya.
Yeah.
Mohon maaf ini saya pakai bahasa Indonesia, mungkin nanti bisa ditranslate oleh Bu Irna. Selamat siang, Pak Alwi ya. Terima kasih atas sharing wawasannya, sangat menarik saya kira. Ini topik yang untuk saya sendiri relatif baru, karena itu juga ini saya pertanyaan yang mungkin dasar ya. Yang pertama terkait penggunaan 3D printing ini dibandingkan dengan produksi secara konvensional, apakah dengan produk 3D printing ini juga bisa menghasilkan karakteristik produk yang sampai tingkat yang tidak hanya bentuk, tapi pada tingkat yang lebih dasar, misalnya bentuk porinya atau bentuk struktur permukaan dan seterusnya. Itu yang pertama. Kemudian, apakah teknologi 3D printing ini juga untuk skalanya, apakah bisa untuk skala besar industri misalnya, atau ini lebih untuk pemodelan di skala yang lebih kecil, skala lab seperti itu? Mungkin itu saja pertanyaan dari saya. Terima kasih.
Okay, thank you, Prof. I just said I can understand. It's fine.
Okay, thank you.
No, it's fine. I do can understand the Indonesian. It just sometimes for me to speak it, then that's going to be a big problem. In terms of the dosage form, it's not just the shape. Like I mentioned, there are a few studies they combine the drug and then make it. They can control the release profile. For example, the Spiratem is the first and actually the only FDA approved tablets in the market. By making it into using the 3D printer, they managed to control the release profile. It means they make it into a fast release profile. This one is because when they're making the 3D tablets, they improve the release profile by making it very porous. So this one is because the technique they use the binder jetting is basically you just glue the powder together layer by layer. So you prepare the first layer, a very thin layer, and then you print. You put the binder to act as a glue, and then you put on the second layer on top of the first layer. So the first and the second layer been glued together by this binder, and so it means you didn't really compress the tablet. So if you compress the tablet, it means the tablet become hardened, then it's going to slow release the tablet. And then the other one, because they just put the powder to be on top of that, is sort of like a you have a sand. So it means if you put a sand in the container, then you can see there is a hole, or it's become porous. So there is a water can seep through the sand. So that's what happened with these tablets. So because it's porous, then you improve the surface area of the tablet. So in a way, you improve the dissolution rate of the tablets. And the other one is because the binder or the glue that they use is water soluble. So when the tablet was in contact with the water, it can disperse straight away. So it's just going to dissolve straight away in the water. So again, when they release, I mean when they dissolve, so it's scattered. It means it's sort of like you just take a powder, a loose powder. So when you take a sort of like a loose powder, you increase the surface area of the drug. So in a way, you improve the solution of the drug itself. And the second one, in terms of the marketability, so far there's only Spiratem. That is the only one 3D printed tablet in the market. So they actually the aim of this technology is at one point you can customize this tablet, because for example, if you go to the hospital, one patient normally they won't just take one tablet. They will take a few drugs together. Which is one of the study done currently done is an ongoing study done by my student. She combined the isoniazid with the vitamin B6, because normally when the patient when the patient taking the isoniazid, then the doctor will prescribe them with the B6 as well, is to prevent the peripheral neuropathy, if I'm not mistaken. So like I said, sometimes when the patient when they take a medicine, they don't just take one type of a medicine. Sometimes they're going to take a couple of different medicines together. So when you apply this technology, then you can customize it based on the patient. So in the future, one tablet can have more than one type of drug in it. So the patient don't have to take a couple of tablets in a day. You just need to take one tablet in a day. But for now, in terms of upscale or that kind of things, so far they only have one 3D printed dosage form in the market. And so there is no new dosage form in the market yet.
Mungkin sedikit saya melanjutkan Pak Awis ya. Jadi dengan kata lain pendekatan 3D printing ini untuk mendukung pengembangan obat yang spesifik untuk orang per orang gitu ya, pasien tertentu gitu ya. Mungkin sama arahnya dengan mungkin pendekatan farmakogenomik artinya obat yang bersifat spesifik untuk tiap orang.
Yes.
Itu seperti itu ya?
Ya, betul.
Ya, terima kasih Prof. Awis dan Bu Erna. Ya.
Thank you, Pak Sabtono, for the question. Maybe a couple more minutes. I think our students are just quite shy to ask their questions. Don't take it too personally.
No, it's fine.
Yeah.
Yeah, actually, let's say you may ask me the question by emailing me if you want after this. Inshallah, I can reply anytime I'm free.
Thank you very much. Would you share your presentation slides with us so that I can share it with the students?
Yeah, sure.
Where then we can click the hyperlink, yeah, and also your email address. I think it's written on the slide, right?
Yeah, it's a last slide.
Yeah. Okay. Thank you. Okay. It looks like there's no more questions, and I think we have covered all the questions, like from me and from Pak Sabtono. Dr. Awis, is there anything else you want to add before we conclude the discussion?
I just want to thank you for inviting me, and then actually it's quite last minute as well. So again, thank you for inviting me and me sharing with a small knowledge that I have regarding the 3D printing with you guys.
Thank you. I'm sure it's not just small knowledge, it's way bigger than we can ask from you. Thank you, everyone. We appreciate you being here in our webinar, especially to Dr. Awis. We have learned a lot about 3D printing in just very short time, and truly it's a priceless experience to listening to your talk. For the attendees, thank you for the active participation for this webinar, and a quick reminder to fill out the attendance form, and that there will be next webinar series tomorrow, Tuesday, 4th of October at 9 to 11 a.m. with the speaker from DKSH Malpen about particle size and stability analysis using DLS technology. Thank you again, and see you in the next webinar.
Thank you.
Terima kasih Dr. Awis atas waktunya.
Thank you, Prof.